Thank you. The talk is yours.
Okay, thank you. I will talk about the experimental characterization of aerosols and droplets that contain viruses in indoor environments. And this is something we've been doing research on for several years here at Lund University in our research group. Since I'm sort of the first one here in this session, I will also have a bit of a general introduction into airborne transmission of respiratory disease and provide a bit of a framework to that at the same time. I could just briefly mention that we are a lot of people doing aerosol research at Lund University. This is from our 50 years anniversary last year on aerosol science. And some of us are working specifically on bioaerosols and infectious disease, and these are sort of the main people around this work. It's Malin Alsed to the left. She just finished her PhD, transmission of infectious bioaerosols, last month. And Sarah with a red bag, she's presently working on sampling COVID viruses in hospitals. And we have Carl-Johan Frenkel, he's a medical doctor in infectious diseases and also wrote his PhD thesis about airborne transmission. Patrik Medstrand, a professor in virology, and Tina and Anders in the corner. So, transmission through inhalation. There are loads of unknowns here, and this is what we've been looking for. And there are many questions in order to understand this. We have to identify sources, transport mechanisms, and well, also a bit of prevention techniques for pathogens in the air. To begin with, for a disease to transmit through the air as droplets or aerosols, there has to be an emission. And there are lots of different sources for viruses in the air. It could be sneeze and cough. That's an obvious source, which everyone is thinking of. But also talking and breathing. If there are viruses on the skin, we shed a lot of skin fragments all the time, around 1 million particles per hour. Textiles that we are surrounding us with, but also other things like flushing toilets could be a huge source of aerosols, pathogenic aerosols. This is a study we published 2 months ago, where you can see particles emitted from a singer. It turned out that especially consonants produce a lot of aerosols. And there is a broad spectrum of particles. Some will be small and transport for a long distance, and some will be larger and rather rapidly sink to the ground. As you can see in the diagram to the right, just normal breathing produces quite a bit of particle. Talking a bit more, loud talking even more, and the higher the volume of our speech, the higher the sound pressure, the more particles will be generated from the respiratory tract. And to the far right in the diagram, you can see that putting on a face mask is a quite efficient way of controlling the source. So that would be much less emissions of especially the larger particles. If we speak specifically a bit about the hospital world, because a lot of the diseases handled at hospitals, we have a concept called aerosol generating procedures, which is very much discussed. And it's various medical procedures that may generate aerosols, and that thus could be a risk of disease transmission, especially for healthcare personnel. It could be intubation, extubation, various procedures in the throat, surgery with high-speed devices, for instance. Not much research on this yet. Also, I can mention here that there is a confusion between medicine and their definition of aerosols and aerosol science and our definition of aerosols. And this is part of the debate about airborne transmission. In aerosol science, which is my field, we used to define aerosol particles as those which are airborne, and that is typically a cutoff diameter of around 100 micrometer. Above this size, the particles will fall down quite quickly. In medicine, there used to be cutoff diameter of 5 micrometers, and from a physics point of view, a 5 micrometer particle will have a settling velocity of around 1 millimeter per second. So it takes 20 minutes to fall 1 meter. So this is not really a droplet that falls down very quickly. But anyway, this is old tradition in medicine. Goes back to tuberculosis research 100 years ago. I think it's slowly changing, and at least some documents and reports in Sweden are updating their definitions here to become more physical. Could also mention in relation to sources that although sneezing produces lots of droplets and virus-containing aerosols, most infected persons do not sneeze all the time, but they breathe all the time. And the amount of viruses from just normal breathing, or even more with talking, could be more than from sneezing, because we do it much more. This is at least shown for influenza, and it's likely to be the same for COVID. Well, this is just one of, which I think one of the best reports on aerosol generating procedures for virus-containing particles. If we continue, after emission, we have to have efficient transport, and a lot of things happen during transport of viruses in the air. They deposit, they are diluted, and dilution is rather rapid, usually. The particles or droplets evaporate. Evaporation rate below 100 micrometers is rather efficient. It's on the time scale of less than seconds. Particles could coagulate and clump together. And this is also moderated by the humidity in the air. So the drier the air, the quicker the evaporation, and the longer the particles will stay airborne. Just this may be obvious, but I say it anyway, that the particle size of the aerosols that contain viruses, that's different and typically larger than the virus size. So with the viruses are around 100 nanometer in diameter, but they are almost always embedded in a lot of other material. So the virus-containing particles are exist in a wide range of particle sizes. When it comes to particle size, we actually do not know much for viruses, and there is a huge lack of experimental data. A lot of the studies that report that they have measured viruses in aerosols or droplets do not report well what kind of particle size they measured. This is more or less the only one, the only study for SARS-CoV-2, where they measured the virus-containing aerosols up to 5 micrometers or something, or 10 in an impactor. And as you can see, it's not easy to find a pattern here. Basically, the viruses are found in all droplet sizes or aerosol sizes. This is for influenza, where we have a bit more data. And actually, quite a bit of studies show that there seems to be a lot of virus particles in the very small sizes, below 1 micrometer. But typically, this particle size is quite difficult to measure experimentally. You need efficient sampling techniques, and far from all sampling techniques used so far for COVID has an ability to measure below 1 micrometer. Also depend on measuring techniques. Some only capture the smallest particles. Some do only capture the droplets. And many papers do not report much on if it's aerosols or droplets or what kind of definition they use. This is 2 different studies on exhaled particles. To the left, you can see a study of exhaled particles from coughing or sneezing. where they used the method focusing mainly on the bigger particles of droplets. And they showed that some patients produced just large droplets. For some patients, about half the group, it was a bimodal size distribution. So they produced a lot of big droplets, but also a mode of droplets where a large part are below 100 micrometer. Below 100 micrometers, these droplets will stay airborne for several seconds, typically evaporate a bit during this time, and thus be able to stay airborne for much longer. To the right is a study we performed quite recently on particle sizes, where we used a method for measuring just below 10 micrometers and exhale droplets. And there are lots of exhale droplets also below 10 micrometers. Below 5 micrometers, that's roughly a cutoff for the size where the particles can reach the whole lung when they are inhaled. And this size range could be of specific interest, I think, for transmission of infectious disease. That's especially if it's a disease that can infect cells in the deep lung, as COVID-19, for instance. Then, continuing from emission to transport, we have viability, and this is a critical parameter. If a disease should be able to transmit through viruses in the air, the viruses have to remain airborne, have to remain viable in the air. And this is another area where we have rather limited data still, especially for COVID. It has been shown that SARS-2 survive in aerosols. There was a study quite quickly in March, I think, this year, showing you can see the diagram in the right corner that these aerosols survive for hours. After 3 hours, about 10% still is viable, according to this study. Recently, there also appeared a study where they were able to culture viruses from patients. So they collected viruses from a patient in the air of a distance of more than 2 meters and put it on a cell culture, and the viruses were replicating. So SARS-2 is able to survive in the air, but there are lots of things we still don't know. We do not know how temperature affects viability. We know that higher temperatures will decrease viability. There is limited data on humidity effects on viability. We have seen from previous viruses that dry air could be beneficial for many pathogens because rapid drying seems to preserve the biological sort of properties better. This is also affected by radiation and nutrition. And if we talk about indoor environment, which I should in this talk, we can say that the building is rather protective for pathogens. Since the building is comfortable for humans, it will also be comfortable for other biological organisms. And we have less UV radiation. We have less chemistry going on. Okay, so we suppose that we have these viruses in the air and that they remain viable and finally inhaled by a different person. Then we have to look a bit into where they deposit. And this was the topic of my PhD thesis, respiratory tract deposition of particle size, aerosol particles. I could talk about this for hours, but I will preserve you from that. But just could say here that, as I said, below 5 micrometers, the particles can reach the whole lung efficiently. Below 10 micrometers, they're inhaled quite easily. Below 100 micrometers, we have at least deposition in the upper airways. The deposition in the lungs increase with physical activity. So high physical activity will mean that we breathe a higher volume of air and increase the amount of pathogens we inhale. It could be moderated or changed by respiratory disease. So many respiratory diseases, such as chronic obstructive pulmonary disease, the third most common cause of death globally. Alters the deposition of inhaled particles. Can also depend on the humidity and hygroscopic properties of the particles. And almost all ambient aerosol particles have hygroscopic material, and this will change the deposition. Okay, I will soon finish. I have just 3 slides more, and I will take them quickly. Does the pathogen reach areas for replication? This is a critical question, but we can say for COVID that it will reach areas where there are cells that can be affected, then can be cleared. The clearance is less efficient if it's very dry in the air because the ciliated cells work less efficiently. Another critical question, is the dose that we receive through the air and aerosols sufficient to cause a disease? Well, we don't know yet. We don't know what the effective dose is for COVID. But we can say that long exposure time, high exposure concentration, and poor ventilation rate will make things worse. Finally, risk factors for aerosol and droplet transmission. Loud environments, not good, because we will increase the amount of particles in the air. Narrow space, not good. Poor ventilation, not good. Better to stay short time in these environments, and it seems that dry air can be a problem. We don't know if the aerosol particles or the bigger droplets are the worst or most of risk for disease transmission. There can be advantages for both. The larger droplets contain more viruses. The smaller ones have longer transport and are more easily inhaled. So, in conclusion, we still lack a lot of information here for transmission of infectious disease through aerosols and droplets, and even for the most simple questions. Okay, did I?
Thank you so much. We already have a question. Thanking first for the talk. Do you possibly know the filtering efficiency of common face masks as a function of aerosol size?
Well, yes and no. It was on one of my slides actually. So if you look at the diagram to the right, here we measured the amount of particles that were exhaled, and also during loud singing, where we have quite high emissions, but wearing a face mask. So when it comes to filter efficiency for filtrating the source, removing the source, particles below 2 micrometers are not filtered very efficiently with a common surgical face mask. I mean, these simple devices. On the other way, it's typically the same way around. But there are face masks which are quite efficiently, and at least the filters capture almost everything. But this is also changed by reality. So having a beard or a loose fitting will make things worse. And without a very sort of advanced mask, there will be substantial leakages.
We have another question. Thank you. And it's about a claim in SVT, Vetenskapsvärld, so the world of science. They say that 1 in 2,000 droplets contain COVID-19 based on a paper they did not actually cite. Do you have any comments on this?
Yes, I saw the claim. Well, I saw this, and I also found the paper, but I haven't read it carefully, I should admit. And I was a bit confused. I mean, it's a very small fraction of all the aerosol particles in the ambient environment that contain viruses. We know that. It's about 1 in 100,000. If we just measure particle numbers, it's a very small fraction that will be virus containing. But well, what the conclusion is, I don't know. It's some of them do contain viruses, and some viruses are enough to cause infection. So, well, a lot of the things we inhale are not infectious, but some is, and we do not need a lot of biological material to get sick.
Jakob, we are out of time. Thank you so much. Please release your screen so that Sophia may start sharing. Thank you. And once again, applause.
Thank you.
While Sophia is starting to set up, let me say that we did have more questions than we had time to answer, and we can go back to these questions when we have the panelist discussion. Thank you for participating, everyone that has been asking questions. Some people asked questions through the question interface, some people asked them through the chat. Both are fine. Perfect. Sophia is just about going into her presentation mode. And Sophia Hober, I hope that I pronounced that correctly, joins us from KTH Sweden. Sophia, perhaps you could say a little bit more about yourself in terms of your background and, you know, sort of the trip you've taken to arrive at where you are. For example, did you start in Sweden? And with that also, I see that you are sharing your slides in edit mode. Perhaps you want to go into presentation mode before we get started.
Is that better?
You may have two screens and you are sharing the second screen in presentation mode because we continue to see the edit mode.
To swap. So right now, is that better?
We continue to see the edit mode. We can read your slides, but we can also.
This is really strange because I don't see that myself. But
if you stay in edit mode, all you need to remember is that you need to go to the next slide in edit mode.
yeah, but if I don't see the edit mode, then it will be hard to. Can you see anything now? I'll stop sharing and then I'll try once again.
Okay.
So now it's edit mode, I guess.
Yes.
So, and now?
Now it's presentation mode. Thank you so much.
Wow, it worked. Good. So thank you for inviting me here. I'm really not working with the things that you are discussing here today, but I hope that it will be of interest anyhow. So my background is actually that I'm professor in molecular biotechnology. So I'm working with the small parts that actually keep this virus together. And normally I'm working with developing things for therapeutics and also diagnostics. And I have not been in the virus part before, but when COVID entered to Sweden, we realized that we need new technologies in order to track this virus. And that's why me and a bunch of my friends went into this research regarding COVID-19. And what we have done is to develop a high throughput method for detecting antibodies in patients or individuals. And we also have been used that for different things and continuously using that. And I will take you on that journey to show how everything started, how we have developed it, and also some glimpse about how we use this so far. And of course, any question is welcome. So many of you have heard about antibody detection, and what struck us early on in March that there were no really good way to measure antibodies in serum, and it seems to be a long time since we have that on the market. So therefore we decided to start to develop it, and since there also was hard to get material at that point of this pandemic, we decided to produce it ourselves here at KTH. But why do you want to know, or why do you want to detect antibodies? That's also something that I think that you are aware of right now. Everyone wants to have antibodies against SARS-CoV-2 because then you can travel around in the world and be happy as nothing has happened. One can think. That at least one way to see it. And we thought that it would be good to ensure who's already had the disease, and that could be used for different ways. We could use it in order to look at it epidemiologically to see how many people that actually have had it, for example, here in Sweden. And using that both to predict what will happen after this moment, but also to plan, for example, who would get the vaccine. And what we also would like to know is how long do you have the antibodies after an infection? Are you at risk to get the infection again? And if so, when? Would it take a couple of years or would it be within 3 months? All of you have seen this nice little ball before. And this is actually the virus. They are really beautiful. Here you have inside the virus a genome, and the genome of this virus is made up by RNA. And it's also circled around a protein that we call nucleocapsid. That's an important protein when it comes to detecting antibodies, because this is used in many of the different assays that you can buy. And this is the one here in the middle. Also, we have different proteins on outside the virus, and here you can see the pink one here, that is called a spike glycoprotein. And this is the protein that the virus actually uses to get into the cells, because the virus is not living, it's a dead thing, and it needs the cells in order to amplify. So it gets into the cell and then it uses our own proteins and enzymes in order to develop and amplify. And when it then has been a lot of different virus particles inside the cell will fall into parts and the virus comes out, and that cycle continues. So when we wanted to do this, we wanted to try many different of these proteins to see which one was most efficient in order to see if we had antibodies, but also which of these would actually be good in order to separate between other viruses and this virus. So we surely know that we detected antibodies against exactly this virus. And you know that there are many different available tests. In the beginning, if you get the virus, you would like to analyze if you have the virus in the nose or in the throat. And there you use the PCR in order to amplify the genome. That's the most sensitive test that is available. Also, there are coming new virus protein detection kits that are much faster, but they are much less sensitive. So in Sweden, it has been decided to go for this DNA amplification. But last week, I think they talked about in EU that they should start to use this kit because it's faster. But I think that that might be dangerous, because if it just detects 8 out of 10 of the viruses, people that has virus, those 2 will be outrunning and infecting new people. So then it's better to have a sensitive test. Also, you can go for detecting IgM or IgG, and that is in the blood. And this is used afterward in order to see if you have had the disease. So if you look at in a time way, first you try to detect the viral antigen, and then you have IgA, IgG, and IgM in your body. And IgG is the one that is actually staying in the body for the longest time. And one of these molecules, median time in the body is about 3 weeks. So in order to keep antibodies for a longer time, the body needs to produce new. You can detect this in a point of care test. You go down to your doctor, and then they take a finger prick and use the blood in order to detect if you have antibodies or not. And there's plenty of these around the world right now. But you can also take a venous blood sample and run that on the lab. Most of the lab detection systems are more efficient than this point of care test. But the good thing with this is that you will have the answer within 15 minutes. And here you have to wait until tomorrow in order to get the answer. So when we started this, we decided to produce the proteins ourselves. And the proteins are all the parts of the virus that we thought that we would be able to detect. So when we started with more than 150 different versions of these virus, and we put them on beads, colored beads. So each color is connected to a specific part of the virus. And then we mix all these beads together with the blood sample. And here you can see that antibodies that detect that specific antigen will bind to that colored bead. And then we can detect the colored bead and how many antibodies that actually are bound to this bead. So one laser signal to detect the color of the bead and another to detect how many antibodies do bind to this specific bead. And here's just a picture, the very first analysis we ran in April, beginning of April, after a development of proteins and such for 4 weeks. And here there were a lot of people that had to stay home during this period of time. And you can see that all the lab is taking part here via Zoom. We have been very good in using Zoom these late months. And here you can see an example on the signal intensity. And what's interesting here that we have positive blood samples, people that we know have had the disease, but we also have negative controls. And you can see when we measure against these pink protein, the spike, you can see that also people that never have seen this virus actually have some kind of binding to the virus. And that goes for all these different antigens or different parts of the virus. And if you look at the MFI signals here, these are normalized against the negative controls in our plate. You can see that most people are on this x-axis, and we have the antigens, the virus parts on this axis. That most of the people that had the disease have also antibodies. The antibodies are detected as dark lilac if there is a lot of antibodies, and lighter if there's not. And if you use the 6 standard deviations from the negative controls means and make the data binary, you can see that there are some antigens here that people that have had the disease do not have antibodies against. And those antigens are not, of course, good to have in this kind of assay because you would like to pick everyone. So up here we have the best antigens, and down here we have antigens that do not deliver as we would like to. And here it looks rather easy. You just pick one of these and then you have yes or no, at least yes. But if we look into the negative samples, those that are not supposed to have antibodies, you can see out of 460 samples here, at least 166 had antibodies against one of these different antigens. And you can see even if we pick the best one, we will have individuals that actually have some kind of binding to this specific antigen, but they have never had the disease. When we looked at this data and discussed, we decided that we need to go for more than one antigen, more than one part of these parts of the virus. And here you can see the sensitivity, which means that how good is this to actually detect antibodies in a serum sample, even if the antibody levels are low. And here is the specificity, how good is to this method or this antigen is diversified between negative and positive samples. And here you can see one of these, we have actually 98, so 2 out of 100 will have the wrong answer. And also this antigen will miss about 4 out of 100. But we, looking at all data, we realized that if we take advantage of 2 different antigens, we could increase the specificity up to 100, but also have a really high sensitivity. So if we look at the Venn diagram here, you can see that out of 331 positive sample, we actually could detect antibodies against both these 2 different antigens. But we miss one guy here. He's an 18-year-old guy, which has no antibodies as far as we can detect. And here we have a 99.7% sensitivity. And if you look in 2090 negative samples, you can see that all individuals actually had antibodies, or we have antibodies, 40 individuals have antibodies against one of them and 89 against the other. But when we combine them, we had 0 individuals here, meaning that we have shown that we have 100% specificity. And this is really amazing if you compare to most of the methods that we have out there right now. We also could see that it takes time to develop antibodies. Here you can see that it's no idea to start measuring before 16 days because still people do develop antibodies. And here we have these young 18 years old that still do not have any antibodies. We went forward to use this method on individuals from Danderyds Hospital that working there. So we started with 2,149 individuals. and test the bloods. We sampled them in the end of April, beginning of May. And during this sampling, we could, when we analyzed the samples, we detected 90% of these employees that actually have had the disease, which was really high at that time. In Stockholm, I think we have 7% having antibodies in the end of April. So this was really high. It looks, or we are convinced that they actually got the disease at work. And here, 9% of those reported that they have had no symptoms at all during this period of time, not even headache. And that's amazing, I think. And this figure is actually quite low compared to other analysis that has been done. For example, in Luxembourg, they detected 30% without symptoms. We could also see that here, that if you have nausea or ageusia or fever, you have a higher odds ratio to have this actual disease. And when we went further in to look at these different individuals, we could see that this was really at the hospital, the infection was really linked to patient contact. So if you had no patient contact, 9% had the disease, while 21% of these with patient contacts got infected. And if we divided these to patient or individuals that have had contact with infected patient and not infected patient, and also looking at different units at the hospital, we could see that the COVID emergency unit and the sorting units, where you sorted COVID patient from those without COVID, they had the highest degree of infection. Also, we could see that the duration of antibodies is not lifelong. In fact, when we sampled these individuals in August, we could see that 90% of those that had antibodies against COVID in May actually didn't have as high levels as they reported out as positive in August. So conclusions from this is that we developed a really high stringent throughput method for serological detecting of antibodies. And by using antigens, 2 antigens instead of 1, we could ensure high specificity and sensitivity. And here I think that you should compare with those that you can buy out there that are normally something between 80 and 90% sensitivity. More than 80,000 samples has been analyzed with this method, and 58,000 individuals has got their results from us. And we could also conclude that we have a large risk working at the hospital to get the disease, and therefore stringent measure in order to protect the people that works there are necessary. Acknowledgements, of course, it's not just me that done this in these 7 months. My lab, together with Tegel and Hedhammar lab here at KTH Albanova, and Peter Nilsson's lab at KTH Sarlife lab, and also Tolin's lab at Danderyds Hospital has been very hardly engaged in this, and we continuously work with these issues and questions that we get during this work, and we have got money from a number of different foundations that I won't go through here. So thank you for listening, and you might have questions. I hope so, at least.
Thank you, Sophia. That was very impressive. I am particularly impressed with the sensitivity and specificity of your instrument. But we have questions, and I have to start with the audience questions, not mine. First question, did you try to test against IgA from airways early in the infection stage?
No, we have only used venous blood in our analysis, and we have detected IgM, but not IgA yet. That, of course, would be interesting. But I think that we need to develop the sample prep quite a lot before taking samples from the mouth and nose.
And the second question that came in, is there any data about the frequency of antibody against different surface proteins?
So we can detect, I can talk about this for hours, but the most stringent surface protein that is on the virus is a spike protein. That seems to develop a high amount of antibodies against the spike, and that's also the antibodies that protect ourselves from the virus to invade the cells. So also we can see that when we take the sample the second time, antibodies against the spike is those that are actually kept for longer time than the rest of the antibodies.
We have time for one final question. If antibodies don't last for long, how long would a typical vaccination have effect?
Yeah, that's a really good question, and I hope that we could start in the evaluation of the vaccines when they come to Sweden here. But they normally you actually get plenty of doses, or at least 2 doses of the vaccine, which in a way remind the immune response to detect the virus proteins. So by adding a booster and a second booster and a third booster, you can keep the antibody response up. So that's a way to handle it. But I think that also here, as we normally see when it comes to antibody against viruses, the antibodies will decrease by time.
Thank you so much once again.
Thank you.
Once again, an applause. I take advantage of the fact that I have sound, so I'll make it louder.
Thank you so much.
I think it's wonderful work you're doing. Etienne, please get ready to share your screen and your slides. Etienne Robert is joining us from the Polytechnique Montréal at Canada. Whenever you're ready.
Good afternoon, everybody. Just give me a second to verify that I have the right presentation. It's the right one.
I can see your screen.
Yes. Am I in the? I'm activating the full screen mode.
I can see it and I can hear you clearly. Thank you.
Perfect. Thank you very much. And thank you for inviting me for this seminar. It's a very good occasion to reconnect with some colleagues at KTH. So I will go right ahead. So first question that I asked myself after I was invited is, what am I doing here? I'm a combustion scientist. And it turns out that I'm a bit of a curious combustion scientist, so I end up stumbling on interesting problems. And if you'll indulge me, I'll give you a little bit of context as to why I started working on acoustic, on aerosols and now bioaerosols. The thing is that I spent a couple of years without access to a combustion lab, and I was afraid of getting bored, so I started a new field of study on acoustic particle manipulation, and that led to projects on the online sorting and sampling of aerosols for real-time airborne pathogen detection. And now I moved back to Montreal a few years ago, and now that I'm in Montreal, I initiated some new research with the Children's Hospital here on the aerosol production around the sink environment. So this project has been going on for about 2 years. And now we also have another project together working on aerosol production in cooling towers. And both the sink environment and cooling towers have been associated with large-scale outbreaks of airborne pathogens here in Montreal and elsewhere in Canada in recent years. And then March happened and COVID entered our lives. So my side project suddenly became very interesting to a lot of people. So I will be discussing a little bit as well what we've been putting together in recent years or in recent months to address the COVID crisis. So a little bit of background to begin with, then I'll be presenting our results on what happens around the sink environment and the experimental technique that we've put together. And then I'll be talking about the 4 main projects that we have going on regarding regarding the spread of aerosol in the context of COVID. So one is with critical care specialists, especially regarding non-invasive ventilation. One is with nose and throat doctors, another with dentists, and a final project with ophthalmologists. So my main interest is polydispersed aerosol in combustion. And I stumbled on this subject when I was finishing my PhD thesis. And what you see on the left here is a quite unique flame that I built in the lab. And what the specks of yellow that you see are soot particles that are growing so big in this flame that they become visible to the naked eye. So electron microscopy imaging of this soot revealed that there's some macro aggregates of carbon nanotubes being built in there. And then I kind of thought, well, it would be nice if we could split it from the rest of the soot, and then you could have a nice way of producing or extracting valuable nanomaterials from otherwise worthless soot. So that's why I stumbled upon acoustic particle manipulation. And then I realized that in liquid phase microfluidics, this technique is very powerful and can do many things like sorting particles according to size or density. And they are very interesting devices that have been built in the last 10 years or so that can do, for instance, what you see on the bottom is a device that can do bandpass filtering. So it can be low pass, high pass, or bandpass. And the way it works is when you have a standing wave between within a resonator, particles will be naturally will experience a force that will move them towards the pressure nodes or antinode, depending on their properties. So you can have very simple resonators, like what you have on the top right, where you come in with a sample and you have a carrier fluid where you can move some of the particles to, and then you can separate them from the rest of the mixture. So the main idea behind my work in this field is, well, I want to find out what the physics authorizes us to do in gases. So in liquids, it's fairly well established, but in gases, there's still a lot of unknown. And my first PhD student at KTH did his PhD on this very subject. And we build resonators that are a bit different than what is done in liquid phase microfluidics, which are typically hundreds of micrometers in size. They're etched in silicon and they're actuated at megahertz frequencies. We worked with electrostatic transducers in the tens of kilohertz range, and we had a channel on the order of about 10 millimeters. So you see the particle stream comes in this rectangular cross-section channel. The transducer is this vibrating membrane on the bottom, and we create the standing wave between this transducer and a reflector. So a bit of samples of results. Our first results were about figuring out what type of separation efficiency we can achieve as a function of the velocity in the channel or the frequency of the actuation. And it turns out that the basic theoretical understanding of the fluid mechanics and of the acoustic radiation force is sufficient in that it can to predict this separation efficiency to some extent, and we achieve quite decent manipulation of particles. And what we did that was a little bit unique is that we were dealing with submicron aerosols. So dealing with submicron aerosol, it kind of becomes interesting when you're dealing with airborne propagation of viruses, and that was one of the applications that we addressed. But when you want to design some specific resonators for a specific task targeting a specific type of particles, then you need a little bit more than these general relationship. You want to be able to predict the force. And for submicron particles, the magnitude of this force is not captured by theoretical models. So we try to validate these models by building a bit of more advanced resonator. Especially we wanted to do a little bit more advanced sampling. So we had this double slit apparatus inserted in our resonator, so we could sample just a few square millimeter of cross section within the channel, and by scanning this around our channel, we were able to measure the displacement of particles with very high precision, and from that extract the force. So the force here, the radiation force, was our unknown, and it's proportional to the particle volume and to the acoustic energy. But there is this phi here, which is the acoustic contrast factor, and the acoustic contrast factor is what is difficult to measure because it will depend on the particle size, but also on the fluids, on the viscosities. It will depend on the acoustic properties of both the gas and the particles. So we measured this down to particles down to about 100 nanometers in size, and we found qualitatively the same relationship or a similar relationship to what the most advanced theories were predicting, but something a bit weird is happening below 150 nanometers. We're still not quite sure what it is, but most likely the physics used in these continuum fluid mechanics models, they do not hold in this molecular regime. But still, it kind of hinted that it was possible to manipulate very small particles with this technique. And as I mentioned just before, we applied this to the problem of sampling aerosol particles. So this was done through the neurosensor project, which was active at KTH from 2012 to 2016. And it was based on the microfluidic detection assay for doing real-time detection of the norovirus, which is the virus responsible for gastroenteritis, or like I think you call it the winter sickness disease in Swedish, if I recall correctly. So these assays, they were based on the use of a quartz microbalance, and a quartz microbalance is just a very tiny resonator, which is functionalized to capture antigens or capture viral proteins, and they are detecting very, very tiny masses in effect. There's different techniques to actuate them, either harmonic or anharmonic, and it's basically sensitive in the picogram range. There's other sensors that are developed, such as surface plasmon resonance based or photonic ring resonators, and all of these they have similar requirements. If you want to do real-time detection of aerosol suspended in air containing the pathogen of interest, then you need to have a reliable way of capturing a lot of these viral particles and sending them into very small liquid volumes. Typically, they deal with hundreds of microliters sample volumes. So we quickly found out or realized in this study that the sampling part was quite challenging. Especially for norovirus, the particles are very small. It's a 30 nanometer virus. The infectious load is very low. So if you want to detect potentially hazardous conditions, you need to be able to detect what a human would breathe or be in contact with in terms of up to only 10 viral particles. The concentration in bodily fluids for norovirus is extremely high. So it turns out that you can generate very small droplets that contain enough virus particles so that only one viral particle can contaminate someone. So it's up to 10 to the 10 genome copies per milliliters of bodily fluids in the case of the norovirus. And it means basically that 1 milliliter could contaminate 1 billion people, which is quite scary when you think about it. At the time, we didn't have any measurement of what was the actual expected virus concentration in air in clinical settings. So we had to do a lot of guesswork. Towards the end of the project, ironically, these results were published, and more ironically, they were published by someone from Quebec City, just not too far from where I work now. So it turns out that in the case of norovirus, you have between 10 and about 1,000 or 2,000 genome copies per cubic meters of air in clinical settings. So that kind of means that if you work with a quartz microbalance and you have detection limits on the order of 1,000 to 100,000 genomes per milliliter in your liquid in the microfluidic system, it means you need to sample like a couple of cubic meters of air, and that takes time. And another problem is that in air you have more than viruses. Actually, you have on the order of 10 billion times more other stuff than viruses. So if you concentrate all of the aerosols within 10 cubic meters into your 100 microliters of liquid that you want to run through your microfluidic system, you end up with slurry. And it will not be very effective to try to detect the viruses in such a high amount of material. So the idea was to try to implement. Sorry, I have some computer issues. Okay. So the idea was try to implement some online sampling and using electrostatics So electrostatics is very useful to useful to capture submicron particles. It's very effective to capture a large amount of them. So this is the basic design that we investigated. So the idea is to use a small volume of liquid electrically connected to the ground potential of the electrostatic system. So there's about 100 microliter in this droplet that it's kind of held by surface tension forces within a 3D printed plastic cage. And you have an airflow in this channel, and the needles here are charging the particles that are then attracted to the droplet. There are other electrostatic samplers that are available on the market, and they simply like sample on the electrically charged surface. The idea is to try to make this approach capable of being directly integrated into the microfluidic system. And since I arrived in Montreal, I also resumed working on this. And the idea is that we want to implement acoustic manipulation stage upstream of the electrostatic charging so that you could charge only the particles of interest and not all of the particles. So mainly the idea is to try to target the particle size that are most likely to carry the virus and remove all of the dust and inert material that is likely to just contaminate your microfluidic system. So now I'll move on a little bit to what has been going on in Montreal in recent years. So the three topics I'll be talking, or the two topics I'll be talking about, is the ongoing project on the sink environment, most specifically the effect of the aerators used on sink faucets on the creation of aerosol particles, and what we've done in recent months on the COVID project. So the cooling tower project is just starting, but it's been motivated by some large-scale Legionella bacteria outbreaks in Quebec in recent years. And that's also another topic where long-term care facilities are especially vulnerable. So what we have at our disposal are the standard tools for aerosol characterization. I don't have a lot of tools that are specialized in bioaerosols, so we rather use and adapt protocols available in the literature to use standard aerosol characterization tools to characterize the bioaerosols that we are dealing with. So the simplest to use is an optical aerosol spectrometer. It's a sampling instrument. It pulls 1.2 liters per minute, and it can detect particle in the 25 or in the 250 nanometer to 35 micron range. A more sensitive equipment for submicron particle is a scanning mobility particle sizer. You can adjust the sampling rate, can go down to 0.3 liters per minute. So that's very convenient if you don't want to perturb a sensitive flow. You can pull just a very small amount of gas, and it's sensitive from 10 to 1,000 nanometers. And we recently acquired a cascade impactor, which is also a sampling instrument. It's sampling at a higher rate, 10 liters per minute, and it separates all of the particles in 14 different stages between 16 nanometers and 10 microns. And we have adapted protocols to extract these particles and do bioanalysis on their biological content. So now we need strategies for seeding the flows to generate our aerosols, especially because part of our objective is to assess the aerosol transport and the efficacy of sampling and strategies or protection strategies. So what we deal most of the time is we deal with inerts or non-biological aerosols. So we can seed with water that we add a known amount of salt in, and then we can analyze the water-based aerosol with our spectrometers or with the cascade impactor. But if we dry the aerosol before we do the analysis, then we analyze only the salt content, and it allows us to find out what the size distribution was as the particle was produced, because small water-based aerosol dry out very quickly in air. And there's two things that you want to know. And you want to know what's the size of the particle when you detect it, but you also want to know what's the size of the particle when it was produced, because that's what will influence largely the transport problems. if we want to avoid this drying out problem, we use DEHS oil, which is a food additive. It's inert, it's safe, but it's also has a lifetime of hours in air instead of seconds or or milliseconds for water. So we can have like stable aerosols using this oil. For biological measurement, we tag our water with a bacteria that has been made or has been modified to exhibit fluorescence. So it allows us to do flow cytometry and culture using this water contaminated with this Pseudomonas bacteria. So for the sink environment, we are interested in particular on the effect of the aerators that are typically used to limit the volume flow of water in faucets. The sink environment has been linked to outbreaks of opportunistic pathogens, Pseudomonas stenotrophos and Serratia bacteria in hospital settings. More in particular, we work with the Children's Hospital here in Montreal. And so we have access to their facilities, and we have done a lot of analysis of what's going on in these sinks. And the aerator has appeared as a particular vector for spread because it has like plastic parts where biofilm is prone to grow. It also is exposed to splashing from the sink itself. So the theory is that the drain acts as the bacteria reservoir, but that splashing in the drain makes the bacteria move towards the aerator where it has an ideal medium to grow. And then every time you turn on the faucet, you aerosolize some of that bacteria. So we have tested 10 models of aerator with very different designs and geometries. All of these are used extensively in hospitals in Quebec and in Canada. So we had a quite significant effort at the beginning to do like a survey of what type of aerators are used. And some of them are just a simple grid and there's no restriction, they're high flow rate. Others are having a more finer grid. Actually, those two images are inverted. I'm sorry, I just noticed that. And others are producing these limited or like a half a dozen of jets to try to further reduce the flow of water. So when evaluating these, we used the two techniques that I mentioned before. If you just look at the total mass of particle produced, two of the aerosols that we tested, they stand out. They produce a lot more aerosols compared to the others. And these are typically the aerosol of the aerated jet type. I'll show you a couple of videos in the coming slides. But what was quite interesting is that when you do the same analysis, but instead of using the aerosol spectrometer with inert particles, you do the microbiological analysis with the cascade impactor and you capture the particles, then it turns out that these two aerators, although they produce a lot of fine particles, they do not necessarily have significantly more live or contaminating bacteria in them. So we did these analysis both with culture and with flow cytometry. And you see that aerator number 3 and number 7, they are kind of in the average. And there's some very big variation, like 2 orders of magnitude between the aerators. So this project is still going on, and the technique that we developed in this are being used now for the cooling tower project. We also investigated if cleaning the aerators change the amount of particles or of bacteria detected in the aerosol, because we always had the same amount of bacteria in the water itself. But we did some tests after hours, days of use, and we did also some tests just after cleaning. And it turns out that yes, if you remove the biofilm that has grown on the plastic part of the aerator, then you bring the concentration down. So the aerosols that are produced, they're not necessarily they're coming all from the water, but there's an effect of accumulation within the aerators. So this is what we are doing now a little bit to understand what is happening. We're trying to develop experimental techniques to characterize more the fluid mechanics origin of these aerosols. I'm gonna try to play the video. Let me know if you see it well. So this is filmed at about So this is film at about 1,000 frames per second, and you see this very aerated jet. The void fraction is more than 50%. And you see these, I don't know if you can see with the resolution, the small particles that are ejected from this jet periodically when the bubbles, just like a champagne bubble arriving at the surface, when it is in contact with the free surface and the interface of liquid between the trapped air and the ambient air is becoming thinner, then surface tension instabilities break out this film and you have the formation of these droplets. So we've developed some image analysis tools to try to bridge the gap between what our aerosol spectrometers can measure, which is up to a couple of tens of microns, and what optical like videos can show us. So here we have an algorithm now that does surface detection and particle tracking, and we can extract the size distribution of what we are producing and from about 50 microns upwards. So we're refining our optical technique to try to make this a little bit more precise. And I also have a student that wants to do the numerical modeling of this to try to study the effect of different water parameter and gas parameters on the appearance of these droplets. And then we arrived finally to what happened in March. So I was already in contact with a couple of medical specialists in the hospitals for the projects that I just showed you. So in March, my phone began to ring a lot, and a lot of people had urgent questions to answer, and that I tried to help as best as I can. So we set up a couple of projects, and the main questions that were coming were, people wanted to know, is the medical procedure that I'm carrying on a routine basis risky or not to generate aerosols? And the second question was, what containment strategies can we implement to protect ourselves and to protect the patients? So as I mentioned in the introduction, we started a couple of projects. The first one and the more advanced was in the critical care unit of the Children's Hospital, which was identified as an overflow hospital for COVID patients. In the end, in the spring, they only had to treat a couple of adults. The rest of the system was able to cope with the load. And right now, they don't have any adults in the Children's Hospital here for the moment. And then we had other project for the ear, nose, and throat doctors that started, especially the endoscopic like visualization of the upper airways. And now we also work with dentists and ophthalmologists. So I'll just briefly review what we've been doing and the results that we had, and then I guess it will be time to open for questions. So it appeared quite early that in the pandemic that SARS-CoV-2 is found in aerosols. So the couple of studies coming out of China in March identified the RNA in ventilation system, which kind of demonstrated that this virus could travel in small aerosols, which is not very surprising. It doesn't tell us if it's a transmission risk in these small aerosols, but it was found in ventilation systems, so tens of meters from the source. And necessarily that means that it's a small aerosol. It's been quantified in clinical setting, the particle size, also in March and also in China. And already in March, we knew that you could find it in a particle as small as 250 nanometers. There was then some in vitro studies in April and May that identified the survival, or not the survival, the infectivity after a few hours in aerosols or on surfaces. And we all saw this, it can stay alive for 3 days on cardboard and 1 day on metal. And then people were starting to wash out their groceries when coming back from the store. So these in vitro studies, they are useful to an extent, but they don't really tell us if it's effective or not in clinical settings. So there was a lot of unknown that remains, and it's still a lot of unknown that remains on the actual role of aerosol transport versus the droplet contamination route. So it's still unclear, but we know, or we can make educated guesses from the reproduction number and the patterns of infection that aerosol is not the main route of transmission. There's a recent review that confirms that we still don't have any documented case of long-range airborne transmission. There's a couple of very good circumstantial evidence that short-range airborne transmission can happen, so a couple of meters, but no clear evidence of long-range transmission. So for the projects underway, we want to characterize the risk associated with aerosol generating procedures. We are using mostly the techniques that I mentioned before for detection. And we also have a project on the decontamination of PPE using hydrogen peroxide vapor. So there's a whole other set of interesting aerosolization problems in this project that I won't be discussing here today. So the objectives is to quickly provide answer and guidelines for professional, and to then go a little bit deeper and understand the underlying physics that are at the origin of the generation of these aerosols. And then in the end, we want to contribute to the development of protective solutions and cleaning protocols. So the first device, so to speak, that we evaluated is a variation of the so-called aerosol boxes. And this was pioneered by a doctor in Taiwan very early in the pandemic. What you see here is a picture taken in the hospital where you have like these boxes are used for patient under non-invasive non-invasive ventilation. And it appeared also quite quickly in the pandemic that using intubation is not the best way to maximize the survival rate of severely ill patients. So quickly, doctors wanted to keep these patients on non-invasive ventilation, which is an overpressured mask over the face, over the mouth and nose, for as long as possible. So these patients, they spend literally weeks on the beds with these high pressure or overpressure masks over their face. And there's a lot of instance where the aerosol production from these masks was quite obvious, when there was, for instance, the accumulation of a fluid at the interface between the mask and the face. And when this interface becomes wet, and it's very obvious that you produce aerosol because you can see the spray on the walls of these boxes. So it quickly became a question of interest to find out, like, are these boxes effective at preventing contamination? So the first design that you see here, it's a variation of this initial design. It has 6 access ports for the hands of the medical specialists. There is a plastic curtain over the chest of the patient. And initially, these boxes were used without suction. So it was just to contain, and there was no airflow or controlled airflow within these boxes other than the ventilation provided to the patient. So what we did is that we used high fidelity mannequins to simulate what's going on in these boxes. So we had an infant model and an adult model. And these were connected to what you see on the floor here, which is a respiration simulator, where we can control the tidal volume, we can control the length and the frequency of the breaths. So we seeded the aerosol coming out of this respiration simulator, and then we looked at the patterns of aerosol within the boxes and around these boxes. We identified, or we investigate 2 clinical situations: spontaneous ventilation, meaning just somebody breathing inside the boxes, and non-invasive ventilation, where you have these masks pushing. Yes, Mario, you have a question, or you want to tell me 5 minutes?
We're a little bit over time. We wanted to leave 10 minutes for questions and answers.
Oh, okay. Sorry.
So I don't want to rush you. I think it's terribly interesting, and nobody has time yet.
I was about to. Well, I'll just scan through then the rest, but so we can have a few minutes for questions. Sorry, I mistimed myself a little bit. So we investigate with or without ventilation or suction in the boxes, and the ports open or closed. And the bottom line, I'll just give you the main lesson, is that actually these aerosol containment, as you might guess, they are aerosol concentrators. So if you don't have suctions, you can actually increase the risk very close to the patient. And for non-invasive ventilation, it was basically the same results. For the ORL doctors, the problem for them came that their procedures were quickly identified as potentially aerosol generating procedures because they're playing with the nose with these endoscopes. And they said, well, you should treat these procedures now as if you were dealing with tuberculosis patients, which means that you have to wait until the ventilation of the room has removed 99.9% of the aerosols. So they went from treating 20 patients a day to treating 3 patients a day. And then we brought them in the lab and we evaluated the amount of aerosol produced during these activities. And it turns out that it's only a sneeze that was significant. Their procedure is the endoscopy here, and we didn't detect anything more than what you would have during normal breathing, basically within our aerosol enclosures. Yes. And then we did some tests for them because they want the patient to have a facial covering when they are doing the procedure. So here you have our patient, and he's coughing without a mask with just a simple cloth over his mouth or a 1-ply face covering, and then it's the same cough, but this time with a surgical mask. And it's just qualitative, but you can clearly see that like a 1-ply doesn't stop a lot of aerosol. They just go through. They go through the 1-ply cloth. It's the same measurement here, but quantitative. The 1-ply remove a lot of material of particles, but still there's a lot more than when you compare to a surgical mask. And finally, the endoscopy, we try to see if we could see a plume coming out, because when they insert the endoscope, they ask the patient to breathe and to use his vocal cords and to breathe through the nose. And we didn't see any plumes of aerosol coming out. So just finally, for dentistry, we're dealing with salt-seeded aerosols, and we have artificial saliva. And we're trying to find out if the aerosols we detect, they are coming from the instrument themselves, or if they are coming, like if they are these aerosols, when they go in the mouth, they have a potential of generating new aerosols that are potentially contaminated. So this is an ongoing project, and these results, they're just hours old, and we measure the aerosols like within 1 feet and 1 meter and away in the room, and we can see that after only 30 seconds of use, we have our spike of aerosol, and it takes a few seconds to dissipate where the patient or where the doctor would be located. Finally, for the ophthalmologists, I think this project has not really started yet, but I'm really curious because we want to see what happens when they do the irrigation of the lacrimal ducts. And you see this Y-shaped duct here. When it's plugged somewhere and they inject the fluid for irrigation, it sprays out the other one. So it has a potential of generating aerosols as well. And that's what we want to find out in the laboratory. So sorry for exceeding my time a little bit. I was just targeting the wrong time on my watch. Sorry.
There's so much material to ask questions.
Yeah,
but I'm going to start. You presented so many projects. Thank you so much. I'm going to start with Stefan Zaleski's question. I'm going to quote. I am not sure I understand the statement about no evidence of long-distance aerosol transmission. Is sitting at 1.5 meters without a mask in a restaurant considered long distance, for example?
Yes, but at 1.5 meter without a mask, you still don't know if it's long-range aerosols or just a very lucky droplet that was projected in the right direction with the right velocity. So if you compare to the norovirus, for instance, that I have a little bit more experience with, there were clear cases of like tens of meter propagation in restaurants, for instance. So I think I know which study Stefan is referring to. It's a study in restaurant in China. But in that study, it was only at the table of the sick individual and at the next table where you had contamination. And you could not rule out that this contamination occurred through droplet or contact. So in the years to come, surely we will have more documented evidence that long-range transmission does occur. But right now, we're just a couple of months, less than a year into this pandemic, and these analyses have not been made yet. So for norovirus, for instance, there was a case in a restaurant in New York in the early 2000s where you had somebody that went through and did a follow-up with every people in that restaurant, and they found that people like several tables over were contaminated, and they were not they did not meet or cross path with the sick individual. And it was actually an event that could have been recorded. It was a vomiting event at the table that was responsible for the spread. So you could know that, oh, well, were you like at the bathroom before the guy vomited or after? And then you had like conclusive evidence. So I personally, I do think this long range transmission does occur. I do believe it's rare, but what I've seen in these very recent review papers is that this is not documented yet conclusively.
Hey, thank you.
Thank you so much. Once again, big applause. We had time only for one question, unfortunately, but of course we have a panel later on.
Yes, so feel free to ask questions offline as well if you have some.
Absolutely. Thank you so much. And please release the screen so that Rajat may share his screen, and he has been getting his presentation ready already. And Rajat Mittal is, we can see your screen and we can see your presentation. Rajat Mittal is joining us from Johns Hopkins in the USA. Thank you, Rajat, for being here.
Thanks for having me here. So I'm going to give a talk on a new mathematical framework for the estimation of the risk of airborne transmission that we've been developing. And just to set the stage for this, the way I thought about this problem is in terms of these knowns, a kind of a state of knowledge, all the way from known knowns to unknown unknowns. And a lot of the previous speakers have kind of spoken about everything that we don't understand about the virus. So hopefully I can kind of put a little kind of framework around that to really try to understand what is it that we understand and what is it that we need to know about airborne transmission of coronavirus. Just now in the Q&A section, we were having questions about the importance of the droplet route versus contact versus airborne transmission. And this audience particularly already knows from the previous talks what these different routes are, but more and more evidence is that the airborne route is actually quite important. It might not be exclusively the route, but it's very, very important for the transmission. And in fact, there's more and more kind of studies that are showing that indeed the contact route might be somewhat exaggerated. In fact, the airborne route might be a lot more important for the large scale spread of the coronavirus. In terms of some discussions about what's the right droplet size, what's the range of airborne transmission. So those are two things that come up all the time: short range versus long range. Large, 100 microns or 5 microns or 10 microns. Ultimately, I think we can kind of eliminate all these controversies by noting that any transmission that involves inhalation should be considered an airborne transmission. So I think that kind of eliminate some of this kind of controversy about what range and what size. So anyway, I'm going to talk about kind of airborne transmission. Previous talks have talked about the size of these droplets, et cetera, et cetera. So I won't get into a whole lot of detail about that, but really kind of get right down to the basics of the kind of the flow physics and the physics involved in transmission. So if you look at kind of the stages of airborne transmission of COVID-19 and any respiratory disease, then basically you have 4 stages to this transmission. One is the generation of droplets inside the infected host. The second stage is the expulsion of these droplets. The third stage is dispersion and transport of these droplets. And then finally the inhalation of the droplets. So these are 4 kind of key stages in this infection process. And one thing that over the last 6 or 8 months when I've been working on this, what I realized was that there are lots of scientists from many different disciplines that are working on this, everything from virology, epidemiology, public health, medicine, physics, engineering, and in fact, the interest from public policy, media, and the general public. But there are very few studies that really start to look at this problem in a holistic manner. It's very difficult to explain to people all the factors involved. It's very difficult to really kind of put your arms around all the different stages and try to understand where are the knowledge gaps, right? Where are the big gaps that need to be plugged? And so that is something that I kind of got really into over the last 6 or 8 months, to try and really come up with a conceptual framework that would be easy to understand, which would be comprehensive, and hopefully allow us to gain some new insights into various aspects of the virus transmission. Now, these are the kind of models, by the way, I'm just putting up here, the kind of conceptual frameworks and modeling frameworks that have been out there. Many of them are based on the very well-known Wells-Riley model, which is a kind of a fully mixed type of an approach to looking at airborne transmission. And these kinds of models, it's very, very difficult to really gain some very quick insight from these models. These models are expressed in mathematically complex forms. And what happened about 6 months ago when we wrote this paper, kind of a review article for JFM, kind of talking about the flow physics of COVID, I started being contacted by a lot of public media, a lot of policymakers, colleagues from psychology, behavioral psychology, and other disciplines, where I found it very, very difficult to really explain to them, all the aspects involved in the transmission. So this is kind of the background story behind this. And so the idea was that to develop some kind of a framework that would have a solid scientific foundation, but also be easy to convey to a very large audience that is obviously very, very interested in COVID-19. So the basic framework that we developed was what we call the contagion airborne transmission inequality. And the basic idea behind this is very simple, is that infection will ensue if the number of inhaled viral particles is larger than the minimum infectious dose for SARS-CoV-2. So that's the basic starting point for this kind of a modeling framework. And this is common to a lot of these deterministic models. And of course, the key now is to understand what determines the left-hand side of this inequality, and what do we know about the right-hand side of this inequality. And so this is the part that we spend some time trying to put everything together. And basically, this is a comprehensive kind of a model that incorporates all what I believe are the important factors in airborne transmission, and they are segregated into 3 parts. The first parenthesis on the left is all the factors associated with the infected host. In the middle is all the factors associated with the environment, which is the transport variables, etc. And then on the right-hand side is all the factors associated with the susceptible person. So let me just quickly go through these factors, and I'll talk about, again, I want to really frame this as a question about what do we know about these factors, where are the big knowledge gaps, and what can we learn from this. So on the left-hand side, of course, most important is the rate of expulsion of droplets from the mouth and nose of the infected person. Second, so by the way, this whole thing is expressed as a bunch of multiplication. So it's a very simple, easy to convey kind of a model. And each one of these F factors is some kind of a fraction. Most of them are less than 1, but some of these numbers can actually be greater than 1. So you have the total number of droplets coming out. FVH is the fractional viral load, which is the average number of virons per expelled droplet. So this tells you really RH dot times FV tells you the total number of virons being expelled. Then you have the face mask. If somebody's wearing a face mask, then a certain fraction gets stopped by the face mask. And then out of the droplets that come out, only a fraction are going to actually aerosolize. The larger droplets are going to fall down. So that's FAH is the aerosolization fraction. Then we talk about the environment. The environment is actually going to transport these these droplets. So only a fraction of the aerosolized droplets are actually going to arrive in the vicinity of the susceptible. So that's that fraction that contains everything about environmental transport. FVV is the fraction of these viruses that get deactivated or remain active during the transport process, and I'll talk about that a little bit. So these are all the variables associated with the environment. Then we get to the susceptible, and susceptible has 3 or 4 variables that are really important. One is, what is the rate at which the susceptible person is actually inhaling the virus particles that surround that person. FMS is the fraction that make it to the mask of the recipient. TS is the time of duration of exposure. And then finally, NC19 is the minimum aerosol dose to start the infection. So these variables now encapsulate every aspect of the transmission in one form or the other. And now I'm going to quickly try to do in this next 5-7 minutes, try to explain kind of what we know and what we don't know. So I tried to kind of put together this diagram of kind of our state of knowledge for the 3 sets of variables: the variables associated with the host, the variables associated with the environment, and the variables associated with the susceptible. And I just want to draw your attention to 2 or 3 variables because I won't have time to go through all these. One is the viral load. So FVH is the viral load, and I think this is one of the biggest unknowns. In fact, I consider this to be an unknown unknown, and I'll tell you why. This is a huge unknown. The rate at which droplets are formed, RH dot, is something that we have a lot of data on, but the uncertainty on this is very, very large. So this is somewhere between a known known and a known unknown. And then on the environmental side, the transport variable, the fraction of aerosolized droplets that actually transport through the air. Of course, we can kind of calculate this. This is based on aerodynamics, it's based on aerosol physics. It is something that we know how to calculate, but it's actually turns out to be still a difficult variable to calculate. So this is also another significant variable. On the side of the susceptible, the biggest kind of unknown variable is actually this minimum infectious dose. And we actually do not know a whole lot about this variable. And so let me talk about these variables that are really below the x-axis, because I think that's where the interesting kind of science is. So very quickly, first I'll talk a little bit about the variables associated with the host. So again, you can skip, there's a lot of information out here, but bottom line is that it's been known for a very long time, since at least the 1930s, that the rate of droplet formation is the key. In fact, there's studies going back, very classic study of Duguid in 1946, that they tried to estimate the number of droplets from sneezes. What we know is that we can estimate these, we can calculate these, we can measure these from experiments, but we also know that there is a huge level of variability in our measurements even today. Every month, a new measurement, a new paper comes out with new measurements, and those new measurements look different from the older measurements. So clearly the phenomena is so complex that our uncertainty level is very high. Earlier on, there was a data set shown on the effect of talking. So it's not just breathing, but it's also talking. And of course, coughing and sneezing has been known for a long time. And then of course, the idea of super spreaders, which is that despite everything else being equal, some people just put out a lot more droplets, sometimes 100 or 1,000 times more droplets than other people. So all these uncertainties really add up to give us a very, very large level of uncertainty in this droplet formation rate. In terms of the viral load, so this is something that, again, Jakob actually talked about this, very interesting. So if we just look at the volume fraction, if we take nasal swabs of people with infections and we take volume fractions, calculate volume fraction of the virus, then the virus actually occupies a very, very small volume fraction of any kind of a nasal swab. And in fact, if you do some back of the envelope calculations, it would imply that a person would only be putting out 1 viron containing aerosol droplet per 6 minutes of breathing. So this is just based on volume fraction estimates. But it turns out that indeed people are putting out a lot more viruses. So this is for SARS-CoV-1, the study by Roman. And it shows, especially out here, that a person with CoV-1 without a mask is putting out about 10,000 virus copies in about 30 minutes of breathing. So clearly there's something that doesn't kind of compute here between what we would estimate from simple volume fraction estimates. So I think this is a huge unknown. The virus is doing something to our respiratory tract that makes it very easy to shed droplets with lots of viruses. Otherwise, the airborne route would not exist. So I think this is a very kind of interesting question mark that needs to be addressed by researchers. Then about the environment, again, this is something that we all understand. The environmental variables, the transport variables, of course, depend on aerodynamic currents, flow fields, the velocity at which the jet is coming out of the person's mouth, et cetera, something we can compute, but we cannot, there's so many different situations that are very difficult to compute and estimate this for all possible situations. the virus viability is something that we actually know a lot about. This is the best variable. We know everything about it. We know the dependency of the survival of this virus on UV, temperature, humidity. So all that, this variable is kind of well known. And then finally, talking about the variables associated with the susceptible. So again, I think the key variable here is this NC19, which is the minimum infectious dose associated with inhaled, kind of an inhaled modality of infection. And again, this was spoken about in previous talks. Our estimates from influenza and SARS-CoV-1 indicate that the minimum infectious dose for an aerosol is anywhere between 100 to about 1,000 virus particles. But there's a huge uncertainty about this. We don't even know if this applies to SARS-CoV-2. The other thing I do want to point out is that the aerosol infectious dose is hundreds of thousands of times smaller than the nasopharyngeal route, which means that any kind of a direct contact, etc., you would require hundreds of thousands of times more virus to generate an infection. So this is further kind of proof that airborne route is actually could be very effective. The nasopharyngeal route is actually very, very ineffective because it requires a very large dose. So given all of this, given all these uncertainties on both the left-hand side of this equation and the right-hand side of this equation, what I claim is that it's very, very difficult for any model to actually estimate absolute risk of transmission. What all of these models can do is actually provide only an estimate of the relative increase or decrease in risk based on behavioral modifications, et cetera, et cetera. So I'll show very quickly one example, and then I know I'm running out of time, is that I was being asked by a number of people that, okay, we know that we need to stay 6 feet apart, but if I go from 6 feet to 12 feet, what happens? Does that reduce my risk exponentially? Does it reduce by square of the distance, et cetera, et cetera? And I realized that I didn't have a simple answer for them. So we actually ended up doing a simulation where we looked at the concentration decay in the droplet kind of dilution rate when you have a plume in an external flow, and basically conducted large eddy simulations with thermal convection, et cetera, all the bells and whistles involved, and estimated what is the rate of decay of concentration of a plume coming out from a person's mouth who's breathing. And bottom line is that we start to get decay rates that, depending on whether it's a hot day or a cold day, the plume will either rise up or kind of fall down. We get slightly different decay rates. Bottom line is that the kind of decay rates that we get are about x to the -1. So approximately the dilution is kind of happening at the rate of 1 over x, where x is the distance. And in fact, that's kind of what comes out from this is that that's approximately a very nice conservative estimate, is that your protection factor increases almost linearly as you increase the distance between two people. So with that, I know I'm kind of running out of time. I do want to leave some time for questions. There's some work that we have done on face masks, where we have shown, again, taking this model, very simple model, and just look at the fact that if one person is wearing a mask versus both people are wearing masks, so bilateral versus unilateral protection. And we took some very nice data from a paper that appeared published by NIST here on various types of face coverings, all the way from surgical masks, all the way to just cotton masks. And the bottom line is that even cotton masks, simple cotton masks, multiply cotton masks, can actually provide protection factors all the way from 4 to about 16, depending on whether only the infected or the host is wearing them, or whether both people are wearing. The other effect of face masks is that face masks not only filter, but they also blunt the jet. So that seems pretty obvious that apart from filtering, a face mask also blunts the jet. In fact, you can see here FLIR imaging, where two people are talking next to each other. In one case, I don't know if you can see this very clearly, but the plume clearly reaches the other person. So there's typical conversation distance between two people. But if you're wearing a face mask, even if the face mask is not filtering, it's actually going to make the plume move above and down. So these are the kind of insights that one can get from this type of a model that we have developed. So again, the model is versatile. It can incorporate other types of models in there, like the Wells-Riley model. But the bottom line is that this model is really designed to kind of recognize that we are not just communicating this to other scientists, we need to communicate this to a much bigger audience. And I think that's very, very important in a pandemic such as COVID-19. So I'll stop right there. I'll put up my colleagues who have been very helpful in this and a paper that was recently published on this. Thank you.
I wish we had more time. I was so enthralled by everything you were presenting. Laszlo has a question, Laszlo Fox from KTH. An infected individual has a much large amount of airway surface liquid than normal, leading to many more droplets. What is your opinion? And there's a follow-up question. What is known about the relation between NC19 and the real number that penetrate into an airway cell? And we have time for one question, unfortunately.
Yeah, so the first question is that, so absolutely, all these viruses are extremely effective in hijacking our respiratory apparatus to make sure that they can propagate themselves very effectively. So there is some data that's coming out that's showing that, in fact, SARS-CoV-2, the kind of congestion that's produced in the lungs, so congestion that's produced in the lungs actually helps the virus propagate itself, because more the congestion, more the droplets, more the mucus, etc. So certainly this RH dot, this factor RH dot, is something that is modified whether a person is infected or not infected. There are studies on flu, people infected by flu that show that before infection and after infection, the rate of droplet generation increases even for normal breathing. So that was kind of I think that was the answer to question number one. Question number two, something about NC19. NC19, what is the, I guess the question is that there's a number that's inhaled and the number that actually deposits inside the lungs, and the two can be different. But ultimately, the way these studies are done, they usually just look at the number inhaled and not necessarily the number deposited. So the NC19 is kind of the infectious dose, which is calculated as how much goes in. Some of it might be ejected back out, but that is not typically incorporated into these calculations. Thank you.
Mario, you're muted.
You see, it was going to happen at some point. So now, thank you so much. And now Tino Weinkauf is joining us from KTH, and Tino is the person who informed me that I was muted. You are ready to present. We can see your slides and the animation as well. Thank you.
Thank you, Mario. Yeah, we're shifting gears a little bit. We're not talking directly about COVID-19 in my talk, but we're more focusing on the analysis of vector fields and flows. If we have such simulations, how do we actually get insight into these data sets? So, and my group and Mario's close and a couple of other people, we're experts in visualizing data, in particular data coming from numerical simulations such as fluid dynamics simulations. Vector fields, flows, they describe motion, and it is the goal and the task of integration-based vector field analysis and visualization to make the motion understandable in these flows and really reveal the insights and the interesting patterns in those data sets. So first of all, we have to distinguish between steady vector fields and unsteady vector fields. We find motion in both of them. First, let's look at a steady vector field. So there would be 2D vectors in a two-dimensional domain or 3D vectors in a three-dimensional domain. You can just describe them with little arrows, as you can see that here. And so you can take a particle and advect it a little bit in the direction of that vector, and that describes some form of motion, right? But of course, in the real world, those little arrows, those vectors would not be static. They would instead move around, change the orientation, change their direction and the velocity as well, like their size, essentially. So we need to distinguish between these kinds of vector fields. The real stuff is really happening in unsteady vector fields. The main fundamental mathematical tool in order to understand motion in vector fields, that's the concept of tangent curves. And they're actually quite simple to describe and define. A tangent curve is a curve that is always tangential to the vector field at every location. Can be described very easily using an ordinary differential equation. You essentially just say, here are my tangent vectors of my curve, and the tangent vector should be identical to the vector field at that same location. The drawing here shows it nicely. Now, while this tool is fundamental in order to understand any motion in steady and unsteady vector fields, when we look at the interpretation of the tangent curve all by itself, we see already that all by itself, this concept will be lacking and will not help us alone. So the interpretation here is it's the path of a massless particle in a steady vector field. And of course, actual particles are not massless. And of course, vector fields or flows outside in the real world are not steady. They are typically unsteady. So let's see how we go about that. Here we do have an unsteady or time-dependent vector field with a particle visualization. I have a two-dimensional vector field here. X and Y are my two spatial dimensions, and I show time in form of an animation. Now I can show the same data set in a so-called space-time diagram where I explicitly include time as a third dimension in the blue axis here. The red and the green axis are still my spatial dimensions that I had earlier on the left side as well. Now blue is my time dimension. Both videos show the same data. They are synchronized. It's the same data and the same visualization, particle visualization, just shown in two different spaces. Now, the right side, the space-time diagram, actually gives me a good opportunity to define what a path line is. A path line is, the motion or is the trajectory of a massless particle in an unsteady flow. So you have a particle trajectory and you follow it through space-time. And then the streamlines that we also often talk about, those are essentially the motions of massless particle in a steady flow. So if you, let's say, choose a particular time step and then just follow the vectors in this time step, then you would come to a streamline. So these lines are of interest, in particular path lines, of course, since they describe really the motion in an unsteady vector field. And streamlines are more similar to the tangent curves we discussed earlier. The ODEs are therefore also a little bit different here. I choose on the left side a certain time step where I would like to have my steady view of my vector field. And here I go with time through time to follow the particle through time. And then I can actually do this using an autonomous ODE by explicitly including time as a state variable. So we go one dimension up, and then you just add the 0 here at the end of your vector field, or the 1 here, and then you get either streamlines on the left side or path lines on the right side. So now you can just take these very simple equations here and compute tangent curves for that. And then you have path or streamlines. So that's very nice. So now we know how to deal with the motion of particles, at least of massless particles, in time-dependent vector fields. What about mass? Once we come to mass and we prescribe some mass to our particles, we call them often inertial particles. And of course, now it can become very, very complicated. Now it depends really on what kind of particles you're talking about, what kind of assumptions you make, and there's so many different ways of operationalizing this. Here I show one particular way, where we have a particle velocity and a position, an unsteady vector field, and then also a gravity vector. And the particles themselves have a response time, how quickly they take up the motion from the vector field, how large they are, what's the density of the particles, and what's the viscosity of the flow. If you put all of that together, you can still come to some equations of motion here that still describe the particle trajectories over time. And you can see that this is different from, indeed, massless particles. Here we have the flow around the delta wing and with massless particles. And then here a gravity has been included. And of course, all these particles sag down, right, and are affected by that gravity. As long as you can describe your inertial particles with any particular system, it doesn't need to be those equations, you are then able to use all the stuff that I'm going to talk about now in the following that allows us to analyze and visualize these kinds of flows. So first of all, just looking at the particle trajectories, just looking at the path lines may not always reveal the interesting structures in the flow. Here I show path lines in a time-dependent vector field behind the cylinder. We would want to see the von Karman vortex street here. Just looking at the path lines, we do not see it. But if we look at the streak lines, then we do. And streak lines are nothing but a set of particles, many particles together that, are all on their individual particle trajectory. But we only look at the front, essentially, of those path lines. So streak lines are very, I wouldn't say closely related to path lines, but they are a structure that comes through particle motion as well. And then you can also nicely visualize them, for example, without all this overlap that you have at the top here, but then just place them in a almost even manner. So in here you see that again, I think very nicely in this particle tracer. This describes actually here in some sense a streak surface. So many streak lines together. You seed your particles at a particular line here, and then you throw out many, many, many particles all at vectum. And you only show the current integration step of your particle trajectory, right? And then you see nicely the von Karman vortex stream. So particle tracing like this is good, just showing the trajectories will not really end well. But what if you want to show the trajectories or the streak lines, right? How can you show lines? Well, in 2 dimensions, this is often very simple. There are methods for placing path lines here on the right side in an even fashion that lay it out very nicely, like you can see it here. So you have an even distribution of those streamlines or path lines, and you can still see any underlying data like the map of Australia. In 3D, this becomes a little bit more difficult. And of course, we're talking largely about 3D. If you start to just show particle trajectories all over the place, then of course, many unimportant ones will hide the important features in the data set. One alternative, what you can do, or one solution to this is to select only those trajectories you're most interested in. For example, here we visualize all the streamlines close to, that are close to, or that are highly curved, that have places where they have a high amount of curvature. And that, of course, reveals the interesting structures in the flow. Another alternative would be you still compute all the particle trajectories, but then you have one way somehow to identify what are the most important ones and what are the least important ones. And based on that, you can then modulate the opacity in a view-dependent manner. So whenever something unimportant hides something important, you only show the important part and fade out the other parts. This has been done in many different ways over several years, and the most latest version here from Irene in Zurich is a very fast method that works not only for lines but also for points and surfaces. Okay. So far for the visualization, it's fine. So far for the visualization of motion in that sense, with the sense of geometry, so that we use particles and lines and surfaces. But sometimes it would also be good to just analyze those flows without really needing to look at these structures in a form of visualization. And for that, we can use finite time Lyapunov exponents, for example. So here the main idea is this. You start 2 particles close to each other and you try to understand when you move forward in time, will they separate from each other? So the distance between them will grow, or will they actually come closer to each other? And here you're looking out for places in the domain where the flow separates. So this is based on the so-called flow map. Let me very quickly describe this. I think this may go a little bit too far, but we'll have a go at it. I have again a space-time diagram. X and Y are my spatial dimensions, and the blue axis is my time dimension. I choose now any point in space-time to start my particle integration, and I go forward in time to do that by a certain amount tau. And that is already my flow map. I map one space-time point to another space-time point. That's the flow map, right? So now the gradient of the flow map is really crucial here in order to understand the behavior of the particles and motion. So you go a little bit up and down here in y direction, left and right in x direction, in order to understand how neighboring particles relate to your center particle. And then, of course, some of them will go farther away, some of them will come closer. And that describes the gradient of the flow map. So it describes the behavior of neighboring particles, denoted here with nabla phi. That is essentially an n times n matrix in a 2D time dependent flow, 2 times 2 matrix, or 3 times 3 matrix in a 3D time dependent flow. Now from that, and I will not go through the math, but it's not too difficult, we essentially derive a scalar field that we call finite time Lyapunov exponent, and that indicates places of flow separation. Where we have high values in that field, we will see particles separating strongly from each other. And here's the most simplistic version of that. We actually have a steady field, so-called double gyre. And here you see those vector fields and those arrows. And you can clearly see that all the particles that would be started here on the left would stay there, and all the ones on the right would stay there. So this is very interesting, of course, in order to understand if you want to see whether particles reach from an infected person to another person, then of course you're interested in places where the flow will fundamentally separate, right? And this is one of those places. Here all the particles go to the left, and here all the particles go to the right. And this is exactly what the scalar field on the right, these finite time Lyapunov exponents, show. T is here the parameter now for how long we integrate those particles. And the longer you integrate, the sharper really the separation gets. Yeah? So from this picture, you can then quickly identify particles from the left and the right are strongly separated. If you do this with really good visualization tools, like my friend Christoph Gard from Kaiserslautern, then you can do this so beautifully as shown here in 2D and in 3D for integrating particles forwards, for integrating particles backwards in time, and so on and so forth. And here is a real world example where this truly becomes important. This is the Monterey Bay in the United States. And here you have Monterey, and any pollution that comes from the city would, of course, potentially go into the open ocean. But then you see indeed these ridges here in the finite time Lyapunov exponent field. So those red curves indicate places of high flow separation. That means the particles from the right, from Monterey, will not have an easy time crossing that barrier and going into the open ocean. So that nicely indicates this barrier. And it's a very good tool in order to understand where particles really go without actually looking at particles and trying to decipher this in a complex three-dimensional and time-dependent setup. Of course, you can also apply this to inertial particles. Here are 2 papers for your convenience. Another thing that is also very interesting, and maybe very interesting for what we discussed earlier, is when you not look so much at how they behave, also how they behave over time, but how they behave with respect to the diameter of the particle. So this goes towards the concept of finite time mass separation. Let me just explain this little figure here, and I think this explains it quite well. Particles have been released all from this location in this data set here, right? And this is the space-time diagram of that. So time is, again, this third dimension that comes up here onto the slide. So they all have been released over here, somewhere here behind this surface, and then they go up in time, and depending on the diameter of the particle, that means depending on its mass, these particle trajectories split into two different sets, essentially. One that goes here on the right side of the green curve. Those are the smaller particles, and then the larger, heavier particles go on the left side of the green curve. So that could also be interesting in order to understand or to analyze certain parameters in your models about the COVID-19 droplets or aerosols. Summary, we discussed integration-based vector field analysis. We distinguish between steady and unsteady flows, massless and inertial particles. particle trajectories are important, like path lines, but also concept based on it, like street lines. And then we can visualize those trajectories, of course. Then we need to take care of what we actually want to visualize, because it is relatively complex, or we go away from the actual geometry. We try to derive scalar fields that help us understanding where these particles separate, for example, FTLE or finite time mass separation. Thank you very much.
Thank you. All right, let's see. Please type your questions now. Here's a question for Tino. Can you comment on the minimum data necessary for such analysis?
The minimum data necessary for such analysis. You want to have, first of all, you want to have a good spatiotemporal resolution in order to actually advect those particles. Advection of particles requires you to interpolate between your sample points. And if you only save every, I don't know, 1,000th time step, then of course, an interpolation between those will be no good. So that is required, a good resolution there. Essentially, the question here is, will any interpolation faithfully reproduce the actual particle trajectory? So that can be figured out. And then, yeah, based on that, I think there's another aspect for how long does the simulation need to run? And that, yeah, depends really on your application question, what you want to find out of how long. So, for example, in the Monterey Bay, this was, of course, a day's worth of data. Whereas in other data sets, maybe also with COVID-19, you're more interested in smaller timescales.
We have, I'm going to say one thing. Right now it's officially the break, but I'm going to take 2 more questions. If you need a break, go ahead and take it now for the participants, but I will take the 2 questions that are up there right now. Are these visualization methods available in any software such as PairView?
They are available in a number of visualization tools that are openly available. PairView is one of them. I'm not sure if finite time Lyapunov is available, but some other things are available there that are shown. And then we also have our own visualization tool called In Vivo. Correct. And I can also put links later. Yeah.
Exactly. We can put links together here with the chat, and also when we document in the recording. The final question before we go to the break. Do you have a recommendation of what software tools to use? It's a similar question for vector field analysis. Yeah. Are there any good open source tools?
I will put links there. Exactly the same answer, actually. Yeah.
Exactly. Thank you so much, Tino, and all the invited speakers in the first session. Round of applause to all of you. Thank you. Let's take a break, and now it's a 13-minute break. Do stand up, stretch, do what you need to do, and then we will see each other again in 13 minutes. Thank you. I will pause the recording.
That would be my pleasure to introduce now the first speaker, the keynote speaker as part of this topic, Dr. Jan De Backer from Fluida. And just to mention that Dr. De Backer has background in aerospace engineering with a PhD degree in aerospace engineering, and he is now the CEO of Fluida since 2007. Please, Dr. De Backer, you can share your presentation with us. Thank you.
Excellent. Thank you so much for the kind introduction. It's really a great pleasure to be here virtually. Maybe one day we'll be able to see each other face to face again, but up until that point, I think this is probably the best alternative that we can have. Can you see the screen just to check?
Yes.
Yeah. Perfect.
Perfect.
Excellent. So yeah, as you know, one of the reasons why we cannot be together in one room is that we're, as you know, currently facing a respiratory pandemic, COVID-19. And I think the pandemic really highlights the need for better understanding of these lung diseases. And maybe some of you are aware of how we look at lung diseases in the practice. It's usually just looking at spirometry, looking at functional tests where we ask the patient to exhale into a machine. We measure what comes out, and that is pretty much the way we typically look at the health of the respiratory system. And we believe that actually that is partially the reason why we are dealing with such a severe consequence of these respiratory pandemics, is because the technology that we're using in the space might not be adequate enough to really understand these diseases very quickly, and also to see what treatments could potentially be applied to mitigate some of these severe consequences. I think the good thing is that we're getting better at it very quickly in this current pandemic, getting better insights, getting better treatments. And as you probably all know, the vaccine is also something that is likely to be there in the next few weeks and months. So what we have been focusing on is using our functional respiratory imaging technology to really understand this disease. The functional respiratory imaging is something that we have been developing over the last 15 years. So we started out quite a while ago. And then really the idea was at the time to see if we could improve the way we are looking at lung diseases. At that time, especially asthma, COPD, lung fibrosis, cystic fibrosis, really to understand the diseases better and to also have better tools to accelerate drug and device development. Because again, you'll see that clinical trials using the conventional endpoints require a very large number of patients and quite a long time to study the intervention to see any efficacy. And we wanted to see if we could use an imaging technique that combines medical imaging with computational fluid dynamics, and we can also accelerate the treatment development. So I'll briefly focus on that in the introduction. What is functional respiratory imaging? And then we'll show you some of the recent results in COVID-19, specifically focusing on the blood vessels, because by now it has become quite clear that the vasculopathy, so the abnormal vascular system in COVID-19 is one of the key drivers for the disease. And we'll show that by looking at the CT scans. We'll compare COVID-19 versus non-COVID-19 acute respiratory distress syndrome. So in the beginning, the physicians thought they were dealing with a typical viral pneumonia, which sometimes leads to ARDS or acute respiratory distress syndrome. We'll show you what the difference was between that thinking and what we see in COVID-19, why it matters. And we'll also look at VQ mismatch, because we've been talking in this webinar about the particle characteristics, particle pathways. So one of the questions is, can we use inhalation medication to treat COVID-19 patients? Is it available throughout the entire course of the disease or only very early on and during the recovery? That's also something we will highlight. But to start also, what is functional respiratory imaging? So it essentially combines high resolution but low dose CT scans with computer-based flow simulations. This is the computational fluid dynamics. And as we've seen also in this webinar, it's been extensively used in aerospace engineering, in the automotive industry, and it's quite well validated. If your input parameters, your boundary conditions are accurate and your settings for the CFD are adequate, then the end results is quite reliable. So the idea is really to understand on a regional level how healthy or diseased is the lung structure and how much does the lung function regionally deviates from normal. And that becomes a sensitive biomarker. If you use it in clinical trials, you can see much more in smaller, shorter clinical trials. And in clinical practice, it really facilitates the matching of the right patient with the right treatment. So we start from these high resolution but low dose CT scans. And the CT scanner is just a conventional CT scanner that's now available in any modern hospital worldwide. So there's no need for the hospitals to buy additional equipment. So it becomes a technique which is quite scalable. We typically take a CT scan after a deep inhalation, so total lung capacity, and another CT scan after a normal exhalation. So these two lung levels are acquired. Then the data is uploaded into the cloud. And then we start to look at the internal structure of the lung. And here we go beyond the somewhat subjective interpretation by a radiologist. So typically a radiologist just visually describes the image, but we actually reconstruct the anatomical structures in 3 dimensions. So we visualize them in 3D, but we also quantify them. So we turn them back into numbers, and then we use these objective numbers to assess either the disease progression or effective interventions. And there's a lot of information that relates to ventilation, perfusion, tissue, and also aerosol deposition characteristics. So for instance, we can look at the lung volumes and the lobe volumes. We can look in great detail at the vasculature, which will be the topic of this talk mainly. We can describe the patient-specific airway calibers and see where they deviate from normal. We can also zoom in on the airway wall itself. So often you see inflammation in the airway wall, so thickening of the airway wall, which can be visualized and quantified by looking at the surrounding structure that makes up the airway wall. And you can typically quantify that down to the level where the smaller airways start, around the 7th to 10th generation. We see a lot of heterogeneity in lung diseases, so we typically report parameters on a lobe of segmental level to really capture the heterogeneity of the disease and or the heterogeneity of the treatment itself. If you smoke a lot, then emphysema becomes an issue, so it's the destruction of regional lung tissue. And so here you can see where the tissue, the dark areas represent the emphysematous bullae, so the destruction of the tissue. And you can see whether there's still some vasculature in that area, still some perfusion, or whether the zone is completely ineffective, pretty much. So if we use inhalation medication, we also want to see where those particles end up. And then what we do is we include the patient-specific upper airway, which is a pretty important part to understand regional deposition. We then connect the inhaler, the MDI, the DPI, the nebulizer, and we simulate the flow and we simulate the particle behavior as a function of the patient-specific inhalation profile. And we also use the CT parameters, the lobe expansion, as boundary conditions. So even though it's a simulation, as you know, it has so much patient-specific input parameters that it becomes almost a measurement. So very close to what you would have with gamma scintigraphy, SPECT-CT, and so on. So this really allows us to understand, okay, where do these inhaled particles end up as a function of their size, as a function of the inhalation profile, as a function of the regional disease? And does this mean that for this particular patient, the inhalation route is still an option to provide the right medication to the right areas in the lungs? So it's quite a comprehensive set of parameters that we can extract from the 2 CT scans that relate to the regional lung structure. So whether it's airway volumes, blood vessel volumes, the walls, etc. Because we have inspiratory, expiratory scans, you can derive a functional measure as well. Internal ventilation, ventilation perfusion resistances, and as mentioned, also the exposure to inhaled particles becomes possible on a personalized level without the need to radio label some of these formulations. Another example of the different visualizations of regional deposition characteristics. And again, sometimes we visualize it in a very similar way as the gamma scintigraphy would be visualized, so that the physicians can then also use it to interpret the deviations from normal in individual patients. Now, if we turn to COVID-19, so what happened early on in the pandemic, when all these reports came out of China, especially describing the CT scans, because there were a lot of CT scans that were taken in the early days during the pandemic in China. We had the typical ground glass opacity, so this is a healthy CT scan, but if you have a viral pneumonia, you would see, and we'll see that in more detail, that the black areas would become gray, light gray, darker gray, which is pretty much the exudate. So there's inflammatory cells that penetrate the alveolar regions, and then instead of being black air, they become gray, more, the Hounsfield units actually increase. But beyond that, they also kind of reported some of the vascular abnormalities, and that really caught our eye. Because at that point in time, we were very much developing algorithms to quantify and visualize the vascular system. So this is an example of a healthy volunteer where you reconstruct the CT scans into 3 dimensions. And then we look at the different sizes of the vessels. So here the red zones indicate the vessels with a cross-sectional area below 5 millimeters squared. The yellow zone is the mid zone between 5 to 10 millimeters squared. And then the blue vessels are the larger vessels with a cross-sectional area above 10 millimeters squared. And if you approach it like this, you can actually see how the blood volume that you see in the CT scan, how that is distributed over the different sizes of the vessels. And interestingly, if you do this in a healthy population, so these are about 108 healthy volunteers, you actually see that regardless of age, gender, height, that distribution seems to be quite the same in that population. So the standard deviation on the signal is quite limited, which kind of makes sense. So if you're healthy, the biology dictates that the blood volume is pretty much distributed over the different generations in quite the same manner. Now, if you looked at the initial 93 COVID patients that we received, and now we have about 2,000, and the signal is very consistent, you actually see that specifically for these smaller vessels, this line dips below the healthy line. So there is a level of vasoconstriction occlusion, especially in these smaller vessels. And then from a fluid dynamic perspective, what happens if you have downstream constriction, but you have an elastic system as the vasculature, you have upstream dilation, so compensatory dilation, pre-stenosis dilation, that you very much see happening here in these mid-sized vessels. So this is focused really on the smaller vessels from 2.5 to about 20 square millimeters. If you look at the entire spectrum of the vascular system, again, you see the vasoconstriction occlusion for these very small vessels, the dilation in the mid-zone, and then the larger vessels tend to return to normal. So the compensation for this constriction seems to be happening within the pulmonary vascular system. So also visually, you can very much appreciate the difference between a healthy subject and then a COVID-19 patient, where you don't see too many red zones anymore. So you do see more of the yellow and the blue, indicating the dilation in those zones driven by downstream obstruction and constriction. If you look at the 3D rotating image, you actually see that the entire lung in this case seems to be affected. And that's quite important, because if you just look at the ground glass opacity, so just that gray appearance where you expect black, that's typically only in a few areas in the lung. But we do see that the vasculopathy, so the abnormal vessels, seem to be present throughout the lung. That's quite an important feature to know, because that really drives the way we are going to treat these patients. So how does this relate to clinical parameters such as the need for oxygen? So what you see here is these BV5, so that's really the category for the small blood vessel volume. So that's how many vessels do you still see in the CT scan with a cross-sectional area below 5 millimeters squared. So you can see again, if you're healthy, about 55% of the vessels that you see in the CT scan are smaller than 5 millimeters squared in cross-sectional area, and the standard deviation on that signal is quite limited. Now in these COVID-19 patients, you see that they're all very much below the healthy level. And then the patients that require more oxygen to remain a certain level of oxygen saturation in the blood, they actually have a significantly lower level of BV5 compared to COVID-19 patients that require somewhat less oxygen. So first indications that if you have very narrow BV5s, we have to give you more oxygen to maintain your saturation level in the blood. And that you see also in this correlation. So this is again the BV5, so the CT-based blood volumes, and then the alveolar arterial oxygen gradient. So this is a measure for VQ mismatch. So you see that for smaller BV5 values, you typically see higher gradients, so more VQ mismatch. And you also see lower oxygen pressure, so it all seems to add up, and it's not just something you see in the scan, but that directly links to the levels of oxygen that you see in the blood. The airway component, also a very important component. So you already visually see that it deviates from healthier. So you will see inflammation, you will see bronchial constriction. And also in these spectrum plots, you see specifically for these smaller airways, you see quite a lot of constriction. So you see a reduction up to 50 to 70% of what you would typically see in a healthy population. The interesting thing is that since we've been doing this for quite a while, 15 years, that we have a database with other lung diseases. So we can start to see, okay, does COVID resemble more of a pulmonary hypertension image? So this is PAH, is a condition, pulmonary arterial hypertension, where indeed the disease obstructs and constricts these smaller vessels. It's quite a lethal disease. It's an orphan indication, luckily, so not too many patients have it. But this again shows that in this known condition where we know there's vasoconstriction occlusion in these small vessels, you also see that the line dips below the healthy line. But you see in COVID-19, it's a little bit more severe, especially for those smaller vessels. So COVID-19 is not pulmonary arterial hypertension, but it has certain features that are similar to this disease, which helps us to see, okay, potentially treatments that could work for PH, do they also potentially work in COVID-19, and do they work in the entire course of the disease or early on versus later? A big debate that is ongoing is this typical ARDS, so the acute respiratory distress syndrome, or is this a special kind of ARDS that we see in the ICU? So maybe to paint a picture a little bit, if you expect to have viral pneumonia, then and you would go on to needing intensive care, you would typically see these patterns in the CT scan. So this is what I was talking about. So instead of black, you start to see these gray zones. So pretty much the inflammatory cells, it's the immune response of the body. The inflammatory cells start to flood the lungs with this exudate, with this liquid. And then what you do is you intubate, you increase the pressure in the lungs to really push those cells back to the other side of the membrane. And that's what you do in a typical viral pneumonia that causes ARDS. And that's fairly well described. So the higher PEEP levels actually are quite good at pushing the liquid back and opening up the lungs again, so you start to see more black. Now, what's very crucial, and a crucial difference between non-COVID ARDS and COVID ARDS, is that in the non-COVID ARDS, we did not see this underlying vascular condition. We did not see the vasoconstriction, vasoocclusion that seems to be so much a hallmark of COVID-19. So here you see what a typical ARDS image looks like. So you see a lot of the lung is missing because it's flooded with the inflammatory cells, so you don't pick it up. And it's very different from healthy. Now, why does this matter? And this is an example, and we just published that in Thorax. So here you see, again, the BV5. So this is this small blood vessel volume fraction, so expressed as a percentage of the total blood volume. And again, here the healthy controls, as you remember, they're around 55%. These are COVID-19 patients, very severe COVID-19 patients, just before they were put on ECMO. And I've seen in the program that the next talk talks about ECMO, so the extracorporeal membrane oxygenation, where you kind of bypass the lungs to oxygenate the blood more artificially. So these are very severe patients. And these are these ARDS patients that don't have COVID. And we took a CT scan at different PEEP levels. So the PEEP level indicates the pressure that you put inside the lung. So what is very important to see that if you increase the PEEP level, you actually further reduce that BV5 and you redistribute the blood to these larger vessels. So this is the BV10, this is the larger side, these are larger vessels, so the larger side of the spectrum. So that means that if you have a patient that you're going to mechanically ventilate, but you don't know there's an underlying vascular condition, and you keep increasing the PEEP levels because you don't get the oxygen level up, and you think you have to put higher pressures, it might be that you actually exacerbate the situation by closing off the blood vessels even more. And we think that is part of the reason why, especially in the first wave of the pandemic, the death rate on ventilators was so high. We saw 60, 70, 80% of the patients actually died after they were put on ventilators. And we believe it's partially because the underlying vascular condition was unknown at that time. Luckily, we're getting better, and I think patients are being maybe not as quickly intubated, and if they were intubated, the PEEP levels are at the lower side. So that seems to be bringing down the death rate, luckily. This is a correlation between this lobar opacification, so we get the more typical way of looking at the CT scan, and then the lobar BV5. You do see that correlation, which seems to be somewhat of a nonlinear correlation, where if you have areas that are more affected by the opacification, so more grayish, then you also have more obstruction constriction of the BV5. However, and that's very important, areas with little or no opacification, so the areas that seem to be fairly normal if you just look at them as a radiologist would typically do, if you then measure the regional blood vessels, you already see significant deviations from normal. That's important, especially if we're thinking about potentially the inhaled route to deliver medication, is that are we dealing with impaired ventilation? Are we dealing with impaired perfusion, or both? And how does that play out on a regional level in those lungs? And this is where this study is quite interesting. So this is a study that we did in collaboration with a center in Brazil, where very early on in the pandemic, they used CT scan to also diagnose the patients, because at that point in time, the PCR tests weren't readily available. And the interesting thing is that they took inspiratory and expiratory scans. And if you do that, you can actually, of course, look at the BV5s, so the lobar blood vessels, these smaller blood vessels, but you can also look at lobar ventilation. So if you look at the expansion of the lung and the individual lobes, and also even the individual voxels, you can see what a regional ventilation pattern looks like. Because as you can imagine, the displacements from expiration to inspiration are driven by the incoming air, driven by the regional ventilation. So just having these two low-dose scans provides us with a means to understand, okay, what is the regional ventilation distribution. And if we do that, if you combine this with the accurate measures of the regional blood vessels, we can start to see, okay, are we dealing with VQ mismatch? And if so, in which direction are we going? And again, we did the same exercise as you've seen before. So we correlated the CT-based opacities, it was the exudate, the edema, with the BV5. And again, the same results, the correlation, but zones that look fairly normal are already affected in terms of their BV5. Now, this was a little bit surprising to me, the following finding. I was thinking that areas with a lot of opacities would also be fairly poorly ventilated. We think if it's filled with liquid, then probably the ventilation is severely impaired, and that doesn't seem to be the case. So it seems that areas with opacities are still fairly well ventilated relative to the other zones in the lung. And that has certain consequences. So what we see here is actually the lobar ventilation versus the lobar perfusion, or a measure of, or a surrogate of perfusion. The VQ matching line, so if you're healthy, you're probably somewhere along this line. If you're above the line, then we're perfusing gasless tissue. That means we're sending blood through areas that do not have a lot of ventilation, so there's not a lot of oxygen coming in to diffuse into the blood. If we're below the line, it's more driven by dead space ventilation. So there's still ventilation in that area, but there's an issue with the vasculature. Either there are no vessels or the uptake is impaired. And you do see that in this particular case in COVID, that most of these zones are characterized by dead space ventilation. So the issue is not so much, are we getting the ventilation in those zones or the oxygen in the zone? It's more what is the reason for the vasculopathy? What is the reason for the abnormalities from the blood vessel side. If you look at some individual patients, so you can then see, okay, where do we see a lot of abnormalities in terms of the blood vessels, and how does that relate to regional ventilation? So in this case, you see a lot of abnormalities in those lower lobes, and they remain quite well ventilated. So that is an insight that for drug developers and device developers could potentially be useful to see, okay, can we do something to help these patients? Another example is, so it's more of a diffuse image here, so more homogeneously distributed, both in terms of the vascular involvement as well as the ventilation. Here, quite a severe case with a lot of impairment, both on the blood vessel side, but the ventilation still seems to be fairly homogeneous. And then in this group, you do see that the patients that were hospitalized had significantly lower BV5s. So time and time again, we see that the BV5, so again, the small blood volume extracted from the CT scan, is quite clinically relevant in terms of understanding things like hospitalization, need for oxygen, and so on. So what could we potentially do about it? So there are some treatments that dilate the vessels, and so inhaled nitric oxide is one of them. We don't have data yet in COVID-19, but we did study INO in COPD-PH, so that's pulmonary hypertension associated with COPD as a lung disease. And there we know that indeed the inhaled nitric oxide dilates the vessels, the well-ventilated areas, these smaller vessels. So here the green indicates vasodilation, and then the red indicates some vasoconstriction. So you do see that you open up these smaller vessels and you redistribute blood from the somewhat larger vessels to these smaller vessels, kind of exactly what you would need in COVID-19. So we're quite confident that if you would give inhaled nitric oxide or inhaled vasodilators, maybe in general, in the early stages of the disease, when the ventilation is still well preserved, that you could potentially mitigate the course of the disease. Here you can appreciate this again, this is COPD-PH, but you see the heterogeneity in the disease, but also in the effect of giving an inhaled drug where you send the particles to the areas that are still well ventilated. So to close off on the topic, so we also looked at COVID-negative patients, and there you actually don't see, again, this underlying vascular element, so the curves are quite different. We do face the issue with false negatives. So you can imagine those swabs, etc. If you don't have measure in the right location, might be that the viral load is not sufficiently high to have a good readout. So also their CT could potentially help in further refining the diagnosis, and especially in light of the treatment selection, indicate what is the relative component of the alveolar airway involvement versus the vascular component. And hopefully that will keep the disease a little bit more under control. So here you see that the COVID-negative typically resembles the healthy more than the COVID-19 positive. So the vascular issues, and it's important to really keep looking at these issues because we are getting an increasingly large number of patients post COVID-19 infection, it's the long COVID or the long haulers as they're called, with a lot of complaints. So complaints about fatigue, hair loss, everything that seems to be somewhat related to vascular system. So if we could potentially mitigate the vascular component early on in the disease, hopefully also the recovery phase could be facilitated and we could prevent some long-term damage, whether it's in the lung or beyond. So a few key takeaways. We do see severe vasoconstriction occlusion of these small blood vessels in COVID-19 that we did not see in the non-COVID-19 ARDS. So that already drives, I think, some of the clinical decision-making. We do see quite a lot of lobes where there is no ground glass opacity or edema inflammation, but they already have a severe level of vasoconstriction, which drives VQ mismatch. So I think what the takeaway from our studies so far is that we really have to understand on an individual level what the phenotype is that we have in front of us, and then optimize the treatment for the individual patient. And that's something that is quite easy to do with a CT scan. So we just introduced this approach in the workflow of the radiology. So it kind of, it's almost smoothly integrated. They get the results back, I think, within 10, 15 minutes. So it becomes something that could really help them to optimize the clinical decision. So we're hopeful. I don't think we're out of the woods yet in terms of this pandemic. I think the next couple of weeks and months will be dark. And there will be a lot of cases. I think there will be tremendous pressure on the hospitals worldwide. But we're hopeful that technology and things like this could contribute to keeping the situation somewhat under control until we move beyond. And I think if we move beyond, when we move beyond, I think we're in a much better position in terms of respiratory innovation to also start treating the non-COVID pulmonary hypertension, the lung fibrosis, the COPD patients more adequately. So thank you very much for your attention. If you have any questions, please feel free.
Thank you very much, Dr. De Backer. Thank you for a very nice presentation, very good insights on capabilities of having functional respiratory imaging as a tool with respect to such disorders. Any questions, please feel free to pose them in the chat or question and answer. Yes, one question with reference to the ability to detect the lung deposition of an aerosol. How far your technique is from regulatory recognition?
It's a good question. So interestingly, the FDA is quite interested in these simulation approaches. So we are submitting the first regulatory files, specifically in the field of generics, for approval. So where CFD is a part of the weighted evidence, together with some PK assessments, some benchtop assessments, generator impactor data. So it's ongoing. I think moving forward, it will be more and more part of files for regulatory acceptance, not as a sole piece of evidence, but as a part of everything. Because as you know, a simulation is a simulation. So it also depends on the boundary conditions. And really what is needed to convince regulators is that these boundary conditions are so, call it patient specific or reliable, that also the simulations are reflective of what would happen in reality. And then it's probably only possible if you have an integration between these clinical measurements, such as the CT, the MRI, etc., and then the computational fluid dynamics, increasingly using AI, deep learning, machine learning to also accelerate those calculations and so on. So I do see a good evolution there, which is stepwise, but it's going into the right direction.
Thank you. Just following up on just what you mentioned with respect to the boundary conditions. Obviously, you are using CT data first of all, and then you are using patient-specific respiratory functions, or to read right in your simulation. And you mentioned that you are very close to a measurement like outcome. Can you comment on this? As you mentioned, the boundary conditions obviously are very, very important, in particular when we talk about fluid mechanics and computational fluid dynamics.
Yeah, absolutely. So I think one of the key issues in these CFD simulations, especially for deposition in the respiratory tract, is really understanding what drives deposition, because that's where you need to put the resources to refine it. And in diseased lungs, deposition is very much driven by the regional ventilation, because as you know, after in the trachea, the flow fairly quickly becomes more laminar. So it's really driven by the regional ventilation. The regional ventilation is driven by the disease. So if you incorporate this disease element in your CFD simulations, in this case, we do that by including the patient-specific ventilation characteristics as boundary conditions, then you see very good agreements with gamma scintigraphy, SPECT-CT, and so on. The other element that is an important part is the inhalation profile, especially if you're using dry powders, because often you need to have fairly high flow rates to break up the particles to get them small enough to penetrate deep into the lung. So that's where also the interaction between what the patient does in reality and what you use in your simulation becomes quite important. That's where we have a lot of patient-specific inhalation profiles that we measured during these clinical trials that could be quite reliable input parameters for CFD.
Thank you. With respect to the drug deposition, what assumptions were made, and how was nitric oxide delivered through inhalation?
Yeah, so nitric oxide is a gas, so that's where
inhalation.
There's not. I mean, we've done that in the past. So we've done the actual multi-phase flow mixing models for CFD, but essentially there again it follows the flow, which for COPDs is very important because COPD, as you might know, is very much characterized by ventilation defects and ventilation heterogeneity. So that's why inhaled nitric oxide is an interesting way of vasodilating the right areas. So you're vasodilating the areas that are still well ventilated. That's important because if you start to dilate areas that are poorly ventilated, you actually make the VQ mismatch worse, and you might see even a drop in blood oxygenation. So in this case with inhaled nitric oxide, you would assume that the vasodilator follows the region of ventilation, which from our measurement it seems to be doing.
And one other question on FRI. What measure techniques were used to extract the blood vessels and alveoli in cases of flooded airways?
So what you the technique to extract the blood
vessels?
So yeah, it's a combination. It's a combination of different techniques. So there's algorithms that look at the so-called vesselness. So it looks for tubular structures, and then it separates that from other structures. So which is the way to separate indeed the vessels from either fibrosis or ground glass opacity that surrounds them. And by doing initially that, by having these labeled scans, you feed that into a deep learning model, a convolutional neural network model, to kind of make it better every time you have more patients coming in. And as I said before, we have a whole range of patients, COPD, cystic fibrosis, ILD, so interstitial lung disease, that also have abnormal vasculature sometimes. So by feeding that into the machine and through deep learning models, it kind of has a whole range, and the performance is quite good, and it's getting better every time.
Right. So this is, I think, with this, you also answered to another question: how is the segmentation of the COVID lungs done, manually or some artificial intelligence, machine learning based technology?
Yeah. So interestingly, if you look at our history, so 15 years ago we started doing this very manually. So I started 15 years ago, really almost manually labeling all these individual airways, and it took me about a week to do a scan. And I didn't know at that point in time that there would be something like deep learning that would automate it. I was hopeful at the time that something would emerge, but we've been doing that for 15 years. So we had a lot of engineers doing these labels. So we have thousands and thousands of scans that are extremely accurately labeled that are now being fed into the deep learning model since, I would say, a year or two. And so these segmentation algorithms have become very powerful, both from the airway side and the blood vessel side. And that is really the future, because a lot of these lung diseases, COVID is a nice example of a lung disease where the underlying vascular condition was unknown and probably caused harm. But that's not so dissimilar from what we see in COPD or ILD, where there's also an underlying vascular condition that we simply don't look at, because the only way to look at it would be a right heart catheterization. So going with a catheter into the heart and measure the local pressures, which nobody ever does in COPD. Of course, if you can do something similar with just an easy CT scan, and you can see what the relative contribution of the vascular issue versus the alveolar airway issue, and you can do it automatically with a turnaround time of 10-15 minutes, then the whole field changes. And then I think you will see a much better treatment of COPD patients, and COPD patients causing a lot of exacerbations and high cost in terms of the overall healthcare cost here in the US, 10 billion a year just for COPD exacerbation. So there's tremendous gains to be made if we can get this technology now into the clinical practice.
Thank you very much. Thank you once again. Thank you for your answers. And I would like to all the other questions will be taken during the panel discussions tomorrow. I would like to introduce our next speaker. It's my pleasure to introduce Dr. Mikael Broman from Karolinska University Hospital here in Stockholm. Dr. Broman, you are free to share your slides with us. Dr. Broman will talk on extracorporeal membrane oxygenation for pulmonary dysfunction. You have the floor, the virtual floor.
Yeah, thank you.
Please.
I'm looking for, where's my, you have my screen here now?
You can either share your, yes.
There it is. Now it's all. Yeah, thanks. Yeah, my name is Mikael Broman. I work at the ECMO Center Karolinska in Stockholm, and I got some kind of vague description of what I was supposed to talk about, and it's more like the clinical setting of what drugs we use in practice. And you also have to know that I'm clinical, I'm on call for ECMO transport and emergencies, so it might be so that I have to sort of just drop out. The presentation is rather short, and maybe I will knock in open doors, of course. There's always a risk for that. You may already have seen what I'm going to present to you. On the left side, you have you see the arrow? You see my point? You're good.
Yes, we see it.
Anatomy, and of course you could inhale nebulized drugs with an open or closed mask, and then the drug, the aerosol, will go all the way down. Here you have the typical sort of picture of a intubated patient. You have an orotracheal tube, and that means that you put your device or you apply your respiratory gases, including maybe the aerosolized drug in the airway that way, and then maybe a few days or weeks into the course of illness, the patient may have a tracheostomy, and you have that on the most right panel. And of course, you can see that dead space is very limited when the tracheostomy has been applied. To the left, you have open mask device for spontaneous breathing. It's open, ambient air pressure, and you may apply your device for nebulization at the sort of inlet of that open mask. Two typical devices for that. You could also have a sort of closed mask, and depending on what kind of ventilator you have, some will allow for quite high leakage, more than 60 liters per minute, far above that concerning some brands. This slide shows the ventilator, and then we have the hose or tubing for that. The nebulizer may be connected close to the interface between technology and patient, or it could be further away. Here is a typical or one type of hose for or tubing for invasive ventilation. This is coaxial type, where you have the inner tube for inhalation limb, I would say, of the tube, and it's connected to the ventilator on the exit and on the outlet of the ventilator. And on the patient side, you may have a moist and bacterial filter, but that's not wise to have the nebulizer to the machinery side of that, of course, because it will be damp and it might trap a lot of what you try to get through. You won't get much through wherever you do. You want to have it on this side. and that's the patient side. And here you can see the patient connected to the machine via the tracheostomy, or the tracheal cannula, actually. Today, a lot of us use this sort of moisturizing device, and that's close to the ventilator often, and then the nebulizer may be applied quite far from the patient. Oh, sorry. And this is the expiration limb. So the air comes from the ventilator this way, over the moisturizing, through the moisturizing device, and then up to the patient that way. This is the expiration limb, and there's a filter for that to protect the expiration valves of that type of ventilator. And it's very good to realize that a lot of what you nebulize might end up and clog that expiration filter within hours or within a few days, depends on what drugs you use, okay? And on this lower left frame, you can see how the nebulizer is attached to the limb of the circuit before the Y piece. The patient is on this side, and here comes the flow from the ventilator, and that's the expiration limb. Here you also can see at the Pacquiao Fisher, the moisturizing device, how the nebulizer is attached. The detail of one type of nebulizer, that's just the Y piece or the connector for the device on the tubing. And here you have detail of how it's placed on the inflow side of the Pacquiao Fisher device. So now I have just 3 more slides for you, and those contain text. And I'm sure that you've heard of, and you already know that the particle size determines where the pharmaceutical agents will be deposited. And if they're too large, they won't reach the alveoli, if that's the target to hit or to reach. If they're too small, they will go in and they will follow the gas out too. So you would aim for a size of those particles of about 1 to 4 micrometers. We may use volatile or gaseous agents as nitric oxide, that was told by Professor De Bacca. Or we may also use anesthetic gases, but those follow the flow. So that's a completely different business. Yeah, as said down here, nitrics and volatile gases, but it's not the laughing gas as we use during surgery. That's another kind of nitric oxide. So what we use commonly or less commonly is saline, and that might just be to dissolve mucus and moist airways. Beta agonists to get bronchodilation, reduce airway resistance. We use anticholinergic drugs to get rid of secretions. Acetylcysteine is to dissolve mucus. Cortisone to reduce inflammation in the windpipes or airways. Less commonly, we may use prostacyclins, and that's with the same aim as nitric oxide. That is to reach the ventilated part of the lungs to get to the vascular side for reduced vascular resistance of the lung, and also to sort of reduce the shunt in the same sequence. You get maybe better oxygenation of the patient's blood, since the blood flow is somewhat more directed towards the ventilated parts of the lung. So you get a better ventilation perfusion match. Then we have some enzymes in cystic fibrosis, for example. This Dornase alpha is maybe used to dissolve and increase the chances to get rid of their very thick mucus. And in the last years, we've also started to inhale antibiotics and antifungal agents, which may provoke or put the staff to some risk. And the bigger problem is when your tubing or the hoses get disconnected. Of course, now in the COVID times, there's a risk for the staff to get exposed to the virus, of course, by aerosol coming out of the airways. But it may also be that some of the agents that we try to give to the patients via the airways may be in the long run toxic to or hazardous to the staff. So what happens if you get disconnection of the tubing to ventilation tubing? The lungs may de-recruit, you get atelectasis, the lungs fall together, and what you have sort of strived and worked for hours, maybe days, to get this lung to slowly recruit and get more sort of air carrying capacity might get lost in just a few seconds. The patient may have an allergy or oversensitivity, or just nauseous substances may create a laryngospasm if you inhale the patient all the way down via an open or closed mask. Otherwise, if the substance comes further down, bronchospasm, asthma, and some of those agents are a bit irritating. And for the staff, as I told you, it's risk for contamination and spreading of contagious disease, but you also expose the staff for dangerous or teratogenic drugs. And amphotericin B, an antifungal agent, is one of those, and it also has a tendency to sort of get stuck into the expiration valve. And those prostacyclins we may use for inhalation to reduce pulmonary vascular resistance and improve the VQ match. Those have a very good tendency to clog that expiration filter, as also one inotropic agent called milrinone that we also may inhale, and that was not on that list on the former slide. So I thank you for this short sort of overview or introduction to what drugs may be used. And yeah, I'm open to any questions.
Thank you very much. Very good presentation on overview on the clinical side. Any questions for Dr. Broman? Did you follow how much drugs are found in the blood over the time?
We monitor quite a lot of, especially antibiotics, but most of those are given IV. So we dose to a sort of plasma concentration target. Those drugs that we inhale concerning antibiotics, antifungals, we don't measure them in plasma or blood concerning concentration, not yet anyway. But the question is very valid.
Right, and with respect to also to medication, it's cortisone. It's used, or
yeah. We don't use it via the airways in the COVID patients, but I would say in generalized use in Sweden, we use it for asthmatics and COPD patients. And depending, time changes, but like 10 years ago, it was very common to give both bronchodilators and also cortisone via the ventilation.
Another question, is the aerosol way used alone or in combination with the IV?
The most common use of the airway is to just target the lungs. But if we have a very severe infection, like Pseudomonas, then we might use both the airway and the IV. So we try to hit the germ both ways, I would say, but the general treatment pathway for the drug concerning infections, that's via the bloodstream.
One other question is regarding if there is any condensed water that you find actually after the Y bifurcation, the Y pipe that you shown, and how much is that?
If sort of the tubing from the Y piece down to the moisturizer, sort of to the side of the patient by the ventilator, if that hangs like you have loops, water locks, those are by nurses' aids, they are emptied. So they pull the water back into, we try not to disconnect the circuit, but it might happen on occasion, most often accidental, of course.
Right. Another question, during those times, and by the way, thank you again for coming to present this and taking your time for doing this. There are obvious challenges that you are facing during those times, and what would be the most challenging aspect with respect to what you are doing now?
I would say
it's different from.
I think it's development of devices that are patient compatible, because they won't work for every patient. But then also that I couldn't say that we have the best drugs to apply via the airways concerning penetration. You know, you get deposit in the airways, in the windpipes. But then how far into the tissue you reach with your antibiotic or antifungal agent is quite interesting, but I don't know of any studies like that. And then if you try to get to the infection both ways, those very sort of heterogeneous lungs we see with atelectasis, partial lung necrosis, and stuff, we might never reach the core of the infection in the most severely sick lungs. So I think there's some development to do, but I haven't read into that too much concerning penetration via the airways.
One last question, maybe before we move on. How is the pathology estimated in absence of direct drug concentration measurements in the blood?
Excuse me, pardon. Could you repeat the question?
How is in the absence of direct drugs concentration measurements in the blood? How is this? I think Marco, you have to clarify for me what you meant there.
Okay, concerning IV drugs, or the drugs we use, and most are IV. We have a lot of methods set up to monitor those by plasma concentration. Those are the common drugs. Most of the antibiotics and quite a few of the antifungal agents we do monitor. But then the few ones that we apply via the airways, the same agents might be given via IV. So what you see is the total effect of the two ways to administer the drug. But if we give colistin via the airways, we have no method to assess that. But we don't have that. It would be the same method if you gave it IV. If you're interested in plasma concentration, because there you may have some references concerning minimal inhibitory concentration for certain drug against a strain of an infectious agent.
Right, and this is quantified in a particular way, or the amount
you
of
have the microbiologist when they the samples we send for culturing, they also make resistance assessments, so they know what concentration that half of the bacteria will die. And from that we know how hard we have to hit concerning the plasma concentration. But that's plasma. We don't know the penetration into tissues. And that's where you want to go if the infection is there. So it's clinic. Does the patient improve? What happens to CRP, procalcitonin, etc.? So different biochemical markers for that. It seems like we are getting closer to infectious control, and we sample a lot. We sample very much.
Let's thank one more time to Dr. Broman. Thank you.
Thanks.
And it's my pleasure to introduce now Professor Stéphane Zaleski from Sorbonne Université, Paris, France. Professor Zaleski, you have the video, please.
Okay, let me share my slides. First of all, thank you very much to KTH and you for inviting me to this great session. I'm really pleased to be here. You can see in the background my university, Sorbonne University, but it turns out I'm not on the streets of Paris now. I'm rather in a little village, but we all show our universities in the background because we are proud of them. This work is also actually done together with Cesar Peretti, who is in Argentina, also in Argentina now. And I'm going to talk about numerical and experimental investigation of atomization. So this is a talk that will be quite impressive, or we will communicate best with fluid mechanicians, but I hope it can also have some interest for people who are interested in the disease, epidemiologists and medical doctors. So I start with a short history of epidemiology on droplets. Pasteur thought that pathogens were transmitted by dust particles. So that's why we vacuum clean our floors. We don't vacuum clean our floors so they are clean. We vacuum clean them so we avoid, at least that's what people did 100 years ago. They vacuum cleaned and waxed the floors to avoid the dust and for hygiene. Then eventually Flug shown that the pathogens were in respiratory droplets. And 30 was a lot of interesting things, such as the distinction between droplets and aerosols or droplet nuclei. Studied tuberculosis, showed that you could just with one droplet, you can infect a pig. And made assumptions about the droplet size distributions, including log normal distributions. And he also introduced this arbitrary 5 micron limit between aerosol and droplets, which is a little harmful to real scientific discourse, at least from the fluid mechanician's point of view. I also want to start this talk with this prophetic paper by Atkinson and Vine. You can see that the paper is quite old, but they already said that it's highly likely that an effective vaccine will not be available during the pandemic's first wave, actually also the second and the third wave now, that hospitals will be too overwhelmed to treat most cases. We'll need to rely, as in 1918, on voluntary and forced social distancing measures to mitigate the spread of the disease, including the closing of schools and churches, elimination of public gatherings, high work absenteeism, and the wearing of masks in public. Well, this was almost prophetic. What it was not foretold is that we can be absent, but still there thanks to technology. So this is from the same paper. They have this, this description, and you see that you can consider all the paths of transmission, and one of them is, cough, sneeze, and inhalation. They didn't write speaking and singing here, but we have to consider everything. I'm not a medical doctor, so I contribute what I can. I will contribute mostly on coughing and sneezing, and I cannot contribute much on talking. Maybe an extension of what I do will be able to do that. It's interesting to look at this droplet ejection mechanism. This is droplet ejection from floating bubbles, like champagne bubbles or bubbles that you can see on the sea surface. They eject both jet drops and film drops. The film drops are those we're interested in because they are very small and they travel very far. Why do they travel far? Because the small droplets are absorbed into the turbulence of the air. One of the speakers this morning already mentioned the toilets, so that's a view of what happens when you flush a toilet. It's quite scary. It also is useful to look at this because you see the clear distinction between large droplets and the parabolic trajectories and the small droplets. Now I want to show you a video by Lydia Borouiba. So I'm going to change screens and show you the video by Lydia. Maybe some of you already seen it. And so you can see a cuff, and so you can see here you see the largest droplets at the small distance. You can see them falling down, and then if you look at larger distances, you will see smaller droplets. And so we're still at the short distance. And Mihai, are you seeing the video okay on your screens? Okay. Here at larger distances of about 1 meter, you see the puff, what you call the puff, that is this kind of round vortex filled with droplets. You can see that it initially goes down, but it slowly, the puff starts inching up. This is because it's warmer, because it has the body temperature. So, and see, it's even clearer that it's going up. Well, the line helps us out too. And so this is part of what is dangerous with these puffs. They go up, so they can hang over your heads in a room and then fall on you. So let me change the screen sharing again. Go back to the PowerPoint. Okay. And so, as I said, the hot air turbulent puff can rise, which is dangerous. This is a view again by Lydia Borouiba, but these are still photographs of a similar sneeze and in a near field. And so you see that the atomization process works by having some so-called sheets. What you see here between these lines are sheets of liquid, ligaments, and then the ligaments break into droplets, as you can see here or there. You have these big droplets and these strings of very small droplets in between them, which is a mark of the non-Newtonian atomization. So we want to simulate what is in this box. Here, maybe we'll represent the mouth as a tube, and this would be maybe the tongue, and that this would be a big blob of liquid, and we want to atomize that. We already have an answer to the question we asked, because Dugwit in 1946 did very nice studies which are hard to do better, in fact. He obtained this droplet size distribution, and he clearly stated that smaller than 10 microns, you shouldn't trust it. So he was followed by Loudon and Roberts in '67. So they have a quite different looking distribution, but again, below 20 microns, you cannot trust it. So if you go above the limit where you can trust things, it's more or less similar. So I've superposed the two distributions. And I don't think it's worth trying to fit it with 2-parameter curves, such as log normal. I fit it with a 1-parameter curve, which is the Pareto distribution, 1/d squared. And 1/d squared is pretty good here. So I think we should aim at simplicity and just keep the 1/d squared instead of the log normal. But here I have tried to fit the log normal anyway, the same way that a lot of authors such as Nikas and others have done. And really the log normal is not convincing. First of all, you cannot trust anything to the left. And these bumps may be log normal, but they could just as well be fluctuations. And now I can discuss this further without reference to Loudon and Roberts. There are other papers such as this graph that 2 speakers have already shown at this workshop. And if you look at this graph, it looks different from what I've been showing. It's bimodal. Why is this bimodal? And I have been showing non-bimodal. Well, first of all, it's volume weighted. That is, it's not n of d, but d cube n of d. So it depresses the small d. Second, it's not a log in the ordinate. Well, still it's bimodal, but if you look at this, my previous plot, first of all, the volume weighted changes the slope completely. So we could have this kind of effect with volume weighted. And second, if you look at Loudon, well, it is bimodal. Question, can you trust this gap? Is it a statistical fluctuation? Well, no, because here you should have about 50 droplets, and the statistical error bars on 50 droplets in a distribution is not that big. So there is really something bimodal happening here in Loudon, but Loudon may have had less particles than Duguid. So eventually I still trust Duguid. I think that bimodal is possible if you look at events such as cough, where you have less droplets, but for events such as sneeze, we have a lot of droplets. And I can come back to this, bimodal is less likely. Anyway, to try to be more precise, we moved to something called the cough machine, and from King, Brock, and Lundell. So it's this device. You have a high pressure vessel here and a layer of liquid. And we solved the Navier-Stokes equations that my former colleagues from KTH know well. We use volume of fluid, octree adaptive grid refinement. I don't have really the time to give the details of the method, and I am already talking for a long time. So let me show you rather the results. So you have here the representation of the walls of the cough machine. Is kind of a tube with square cross section. And the simulation done by Raphael Villiers and César Peretti. Here the colors are the speed of the particles. We only see the ejected particles here, not the film. This is the geometry. If we take the dimensions of this geometry, we can arrive at the air Reynolds number of 36,000, which seems too large. If we use the octree grid, the grid size is 20 microns, and with a grid size of 20 microns, you are below the Kolmogorov length scale of 24 microns. So the simulation is really a direct numerical simulation. It's the biggest direct numerical simulation ever of atomization. The previous one by Ling was at a Reynolds number of 8,000. So we have gained a factor of more than 4 in Reynolds number. And it's a pretty fast event. It's a 30 meters per second event. And this is what happens. So here the particles are colored by their velocity. And if this is another view. So if we look first on the right here, we see a lot of particles which are about 30 meters per second at the velocity of the airflow. And so these are very fast droplets that go much faster than the puff that you saw in Lydia's movie, because in the puff, the velocity was typically more like 1 meter per second. So the huge reduction in the velocity of the puff compared to the velocity of the airflow. So these are particles which will go very fast away from that, and they will slow down eventually as they exit. And here we have this region here, which I have zoomed here, and we can further zoom. You see a kind of zoo of ligaments, and you see these bigger structures here called Taylor-Kulik rims. And it's really the distribution of these ligament sizes that leads to the wide distribution, non-bimodal distribution. If you had only one type of ligament with one size, you could have droplets from the ligaments or the rims, which will give you one mode, and the droplets from the films will give you another mode, and you could have bimodal. But here it's not possible to have bimodal. We can also color particles by their size, so we can identify the largest droplets that escape. It's a random event that you get some very large droplets. We can look at the vorticity, and here it's projected in the walls. You see all this very fine turbulent structure, but you don't see a large vertical structure. You don't see the puff yet, and that's probably because we're not far away from the object yet, and so the puff has not had time to form. We can look at the velocity at which this big wave that you see is progressing by taking a cut. So it progresses at the Dimotakis velocity, which was well known, which is a 1 meter per second here. So it means that you eventually will need more like 30 or 50 milliseconds to get out here at the exit. So the simulation should be much longer, but we already spent 1 million hours on supercomputers. And I think that's enough for the purpose we have here. So what is our purpose? Well, we want to know whether we get roughly the same PDFs as in the experiments or in other atomization simulations. And yes, we get that kind of PDFs. It seems to be well converged. These vertical lines are 4 times the grid size, 4 times delta X. And it shows that after 4 times delta x is converged and the bins are well represented. You shouldn't trust here, whatever is here on the right, there's only 1 droplet here, so it's statistically insignificant. But starting from here, you have 5 droplets and it's significant. You can plot this, it's what I call the log normal coordinates. So you have the Pareto here and the log normal. The log normal seems good, but there is a tendency as you refine the grid to shift the PDF for smaller size. So the log normal is not stable when you refine the grid. If you look at what happens when you refine the grid, you can see here at the very small sizes, the number of droplets increases. The green one here is the converged results. So you see how it increases rapidly. So you cannot trust anything here to the left. The parabolic shape is sort of an illusion. What is the perspective of that from here? Well, we want to follow particle motions over much larger distances, treating particles as Lagrangian point particles. It's a little bit like the particle tracking of flow that was shown earlier today, and subject to cooling and evaporation. The initial conditions for this Lagrangian particle tracking will be the output of these DNS simulations. Or what we could do is also use the same code, Basilisk, but then perform inside the same simulation a volume of fluid to LPP conversion. So this is a more sophisticated technique. For the LPP simulation, I will show you on conclusion some simulation by Chang and others. Another perspective to think more deeply about the viral load in each droplet size class. How much virus do you have in the 1 millimeter droplets and how much virus in the 10 micron droplets? Well, this is not obvious. This may not be proportional to volume, but the virus could be rather on the surface of the object. So maybe the viral load could be at least partially proportional to the surface of the droplet and not the volume. This is, I think, something we should think about. So to conclude, this is the image from Chang and others. So you see the humidity and also see the particle trajectories. You can see here the 100 micron droplets, they can fall very quickly or they can fall very far away. So you cannot conclude because the droplet has a given size that it goes over a given distance. I mean, this is not and you need to think in terms of statistics and in terms of what you want to study, of what where is the other person and can it reach him or her. Thank you.
Thank you very much. Thank you very much, Stefan, for this very nice presentation. We have a few questions, obviously, as usual, and one of them: how important is the actual geometry of mouth and the motion of the tongue that is to determine the initial boundary conditions? How essential are those?
I think this is very important. I think that the geometry of the mouth will be very important because you could really affect the mechanisms, and you can have mechanisms that, for example, make continually change the distribution. But we don't have yet examples of that. We should try. We should. One of the things we will do soon is to just make the channel either longer or shorter, and there will be experiments in by Palaf Kent and Detlef Loos and others that match our simulations with the same tube. So we'll just change the length. That's the first geometry will change.
Thank you. Can you comment on how is the volume of fluid data injected into the LPP and the other way around, vice versa?
Okay, so we can identify particles. Okay, so we can color them, identify them. So once we identify a particle, we put a Lagrangian particle at its center of mass, and we carry on. If this Lagrangian particle arrives in a region where we want to resolve things again, for example, if it arrives near a surface and we want to see the impact on the surface in detail, then we transfer this Lagrangian point mass to a volume of fluid patch.
Can you comment?
We try to take. Sorry.
No, please go ahead.
Yes, one of the issues when you do that, but it's more of fluid mechanicians hobby. we try to keep track accurately of the momentum, because we remove momentum, we add momentum again, and we have to make sure that we keep the correct balance of momentum when we do that.
Right. So can you comment on the Weber number range for those particles?
Yes. Basically, this is a very high Weber number. 700 is very, very high, and we probably it's very difficult to do simulations which are well converged, well behaved for any Weber number larger than 200 or 300. So we are really on the verge of what is impossible to do. Nevertheless, the simulations are stable, and we don't see any spurious deformations. So I think we are okay.
Let's thank one more time to our last speaker. Thank you, Professor Zaleski. Thank you, Stefan. It's my pleasure now to introduce, last but not least, Professor Staffan Lundström from Luleå Technical University to talk about the effect of face masks during airborne pandemics from fluid mechanical aspects, a collaboration between 4 universities in Sweden. Please, Staffan, you have the possibility now to share your screen, your presentation.
Right, thank you very much, Mia. So I hope you can see my screen.
Yes.
And yeah, thank you for inviting me. And I will then present a project that has just started, and I will continue, yet another year. So instead of presenting results, that's most of these presentations today have done very nicely, I will rather talk about which tools we will use and maybe some initial results. And I can also say by this first slide, I've probably broken all kind of rules of logos from our universities already now. Anyway, the team is from then 4 universities, and all groups we can say are in fluid mechanics: Chalmers, KTH, LTU, and Lund University. And I pointed out that we have Johan Restedt as an important person, because he initiated this collaboration on a little bit larger scale, you can say. And this is just one project, and we can also say, I can also say that this comes from collaboration done within CMUF, which is about multi-phase flow in industry, Swedish industry. Right. The purpose with this project then, that is sponsored by the Swedish Research Council, is to reveal how liquid particles behave close to people, wearing face masks or not, when they inhale, exhale, and this includes cough and sneeze. So it's a really huge perspective, at least to start with here, what we have said. And the goal within 1 year then is to, from a fluid mechanical point of view, be able to give guidelines on the use of face masks to prevent spreading of virus such as COVID-19. So again, I pointed out we are fluid mechanical people that do this research. The research is then divided into 4 work packages. One is dissemination, which is led by me. Probably this is why I talk today. We have a work package 2 led by Susong Bai from Lund, and that is simulation of droplet flow dynamics. As I said at the beginning, we will look to droplets or spreading near to the mouth or face of people, while actually this project we go further and see in a larger environment where do the droplets end up. The work package 3 is led by Sradjan Sasic at Chalmers, and that's about also modeling droplet and aerosol transportation through mask material. So we go really deep into the geometry of different masks. And then we have work package for experimental validation, which is led by Mika Sjödahl here in Luleå and Ramis Örlü at KTH. And these persons, they also form the kind of steering group that meet quite frequently. Okay, regarding dissemination, we have a web page. We have informed community on progress in different channels. Myself, I was in TV4 actually before the project started, and I showed how you can put out the candle, and that's easy to do that by exhaling than inhaling, for instance. A little bit exemplifying the problem, the big, the quite complex flow situation close to the mouth, especially if then you have also face masks. We also say that we will have an open workshop next autumn, so within 1 year, and we aim also to publish this work as much as possible. So work package 2, simulation of droplet flow dynamics. Here the objective is to see the effect of face masks on safety distance, droplet distribution, and droplet vapor dynamics. And as I said, we then go to scenarios like office rooms, buses, or it could also be shops and such environments. And then also look at the impact on ventilation, strength, and directions, and the effect of ambient air temperature and humidity. The collaboration within the consortium is on the baseline experiment cases, setting up the boundary conditions for these simulations, and also the non-Newtonian fluid effects from that Chalmers will simulate in their model then. Here are still some preliminary results. There has been a scenario study on a box with the size about 3 by 3 by 3 meters. And this is just how it could look like then, how the particles move. But there has also been a validation study validated with the experiments done by other authors, and you can see that penetration distance done by this model, simulation model done in Lund is, it's really good. Work package 3 is droplet and aerosol transportation through mask materials, and leader there is Sradjan Sasic, as I said before, in Chalmers. And the objective is to formulate a comprehensive rheological description of complex fluids. So here we include non-Newtonian behavior, and these are fluids then expelled from nose and mouth in the case of various inhalation expiratory effects. Then the study numerically the spreading and absorption of these complex fluids and aerosol in filter layers of protective masks. And then also not only to be passive to look at it, but also propose innovative designs of these face masks. So the method used is to have a multi-lattice Boltzmann framework, then with the center potential method to reproduce the interface of droplets. We also plan to do conventional CFD of aerosols, where we just let the particles follow the flow. The fibrous microstructure will be delivered from X-ray tomography here at LTU, and there will be focus on the following effects: then microstructure of fibrous mass materials, wettability properties of mass materials, and then this non-Newtonian behavior of complex fluids during expiratory events. Then also aerosol transport through mask materials. So here are examples of results, and that is spreading of and absorption of droplets in real mask materials. So here we have also showing that coalescence of droplets included in these kind of models. And examples of measured quantities could be permeability, probability of droplets as shown in this graph, the frequency of droplets for the positions within the masks, and also how these droplets coagulate or intercept, as shown in the previous slide. Right, we go to work package 4, and here we have the experimental validation, and here are also some preliminary measurements. Here we show 2 measurements technique, that is interferometric particle imaging, IPI, and that is the middle picture. And with this method, we can measure how particles move, but also their size by this defocus image of particles. And as said here, the number of fringes is proportional to the size then. And here we can look at particle sizing from 5 microns to 10 to 100 micrometer. But of course, the window that we look at is quite small. Another method that we will use is shadow graph imaging, and that's a traditional method. And then we can look at larger particles. And what we see here is that this has been mentioned before in this workshop. Somebody is saying P. That is a constant that releases a lot of particles from the lips. So like cheering on Liverpool or PT or any other football team is maybe, we shouldn't do, for instance. Okay, I move forward. There is also been do experiments at KTH, and here are some preliminary flow visualizations on a larger scale, showing with smoke people that, or a model person that exhale without mask, with laser light sheets, and then also smoke. And then also a more interesting inhaling with mask, then you don't really get any of this smoke going in, while exhaling you have a really large effect. Especially what you can see here is that, and that was also commented on here at the first presentation, that it's very important that the mask sits tight if it should be effectful. Right, the plan activities work package 2 is still to really look more into what happens close to the mouth and nose when you wear a face mask or not. So this kind of measurements that I showed, the IPI measurements for baseline statistics that will go to the simulations. And measurements using artificial generated cases. Has not been decided exactly which at this point. And then estimate face mask performance by measurements before and after a face mask. And similar measurements will then be done at KTH, complementary and similar. So this actually finish off my presentation. I will just show a movie that is generated by X-ray tomography, and that would show how, the particle, if you are a particle, how you move through a face mask. This is done by Fredrik Forsberg. I must go back now. I think maybe this will happen as I wanted.
Yes.
Oh. Okay. Yes, thank you.
Thank you very much, Stefan. Thank you. It was a very nice overview of current some other current research related with COVID-19. And any questions for Stefan? We have a few questions actually. Can you comment on taking into account condensation and evaporation? Is this something that this project will consider?
Yeah, we are looking into that, and that would be in the simulations done in Lund. They have not included that yet in their model, as I know, but this will be considered, yes, and that will also be considered in the experiments. What kind of humidity we would have and temperature. But that is still quite short distances that we will look at in the experiments. So it will rather be in the simulations in Lund. I don't know. I think Johan Restad is online. Maybe he could comment on that.
He for sure he can comment, and I can read his comment if he's commenting, if he'll comment it via text. One other question, while maybe somebody will answer from Lund. One other question is, does the experimental validation performed with physical experiments, does it have a quantitative validation method, or is based on quantitative validation method, or it's just a qualitative visualization?
No, it will be quantitative measurements of velocities and also of the size of the particles, for instance. So of course, yes, it will be quantitative measurements. Yeah, I think that is yeah what we want to have. Yeah.
How about the mask sizes? Obviously, there are so many different brands. Also, mask sizes, pore size. Linked with those different brands.
We will look at two different face masks that are commercial at least, and then one that is this kind of bandana or something that you just put with one sheet. It's a little bit difficult, that one, because of course what we will do is that we will, it's already done right now. We will map it with X-ray tomography, so we'll know what we measure on. But of course, these kind of materials can be very different.
Yes, thank you, Stefan. Do you consider also the efficiency decay with the usage of the mask?
Yeah, we could do that. I think this is this is something that a little bit what Radjan and Chalmers are into, that the permeability will cause decrease, while if you get a lot of fluid particles within the mask, so this is something we can consider. Then it's of course there is limited time, but something that we have talked about and something that we would like to do.
Thank you very much, Stefan. I think that our time is almost up, right, for today. Really nice video also. I think that the entire panel agreed on that. As I sit here, our applauses. And yes, we have also a comment from Lund University. Thank you, Johan, for having this comment here. Yes, the intention is to include condensation and evaporation in the simulations at some point. The group in Lund is working on those aspects. Thank you, Johan, again. And let's see. I think one other thing is that one other question, maybe that last question that will take it today. City reconstruction has its own accuracy, which is difficult to go below 2-3 microns to quantify those filters, right? How accurate in the simulations can be this with respect to the smaller droplets?
Which was the question? Was it the simulations on
if we are using CT reconstruction to
right.
to reconstruct the, let's say, the geometry and the fibers and everything, and this is coming with its own accuracy.
Right.
Question is how accurate then the in the simulations.
I can answer at least the first question is about what is the accuracy of the CT scan, and I think maximum accuracy will be right 0.5 micron, but these scans that are ongoing right now are done with 2 micrometers, and then there is an additional, as is pointed out here, there's additional inaccuracy while doing the simulations. I cannot really answer on that one at this moment.
Thank you very much. Let's thank one more time to all our speakers today. Thank you. And I would like to actually really encourage you to also, I know that your time is also precious, obviously, and please try your best to join also the panel discussions for 1 hour tomorrow. And that will be, I think, a very nice, let's say, closure to this webinar. And I would like also to just mention that tomorrow we'll start at 9 o'clock in the morning, Stockholm's time, and we have very nice presentations. The session will be chaired by my colleague Lisa Prahl-Wittberg, and we have also keynote presentations with respect to airway inflammation in chronic airway diseases. So talks from Karolinska University Hospital, from University of Parma, from Göttingen AB with impact on ventilators, and obviously from KTH, Professor Laszlo Fuchs. And then we'll have the panel discussions for about an hour. I would like to thank you, everybody present today, also in particular to today's speakers, but also to the audience. I hope to see all of you tomorrow at 9 o'clock. Thank you again.
And thank you, Mihai.
Thank you, Mihai.
Thank you, Mario. Thank you, Lisa. Thank you all.
Thank you very much.
Thank you.
All right, let's stay perhaps for 2 minutes. Reflect on anything we could do better. And this is mostly for the organizers. The panelists are welcome to stay or leave, and the attendees, again, you are welcome to stay or leave. Mm-hmm. It's not that we're going to discuss some secret matters.
I just want to thank you again for inviting me, and unfortunately tomorrow I don't think I'll wake up at 3 in the morning to attend the session.
We totally understand.
I would have liked to attend. If I have insomnia problem, I'll join the discussion. It's 5 a.m. here, so
we understand. Thank you so much for your presentation. It was fantastic. I wish we could have had more time for it.
Yeah, and it's not a matter of going up early. You just need to stay up late.
Yeah, I was going to say that at 3 a.m. I may consider staying awake from the day before.
I kind of did it in the last few days and few weeks, a bit too often for my taste. So
yeah, no, that takes a toll. And also,
yes, thank you for the good words for my presentation. I would have liked to polish it a bit more. I just had time to put everything together and not much for optimizing. So
I was just going to mention that I can see how you're not sleeping. Everybody wants data from you. Everybody wants models and answers.
Yeah, it's
Stefan will be with you in a second.
It's tough not to take it too personally as well. I've been working with these people for a few years, and like in April they were really, like, you could feel a lot of uncertainty in their questions, and try to contribute a little bit, and hopefully it will lead also to a publishable data set and more understanding in the long term.
Will you be able to join the panel in which is towards the middle of the day? Still early for you.
You mean tomorrow? I would like to, but I think it's I'm looking at the schedule, and
yeah,
it would be it would be 5. If it's 11 your time, right? So yeah,
right.
that would be 5 in the morning for me.
No,
I don't know. Unlikely.
No, just take your rest. The questions, I shared a document.
Don't hesitate to share questions if some come and are relevant for on my side. And there's a lot of things that I didn't have time to talk about on my side as well. And we made some funding requests for hardware to have like a 0.5 meter diameter Schlieren system and things like that to be able to follow plumes a little bit better. So there might be some interesting possibilities in the future.
I just posted, I posted to everybody, but no, I posted only to Mihai. So I'm going to post only to the panelists. I created a Google Doc with the questions, and the link that I shared allows you to edit it. And we have a few panelists who have already answered the questions, in preparation for the panel. So you could answer, if the questions make sense to you and you think they're important enough, you could include your answer, and we'll go ahead and represent you tomorrow during the panel.
Perfect. Thank you very much.
Thank you, Stefan, for taking also and answering to those questions right away. It was nice. Etienne, it was very nice seeing you. Please send my regards to the family.
Yeah, same.
Stefan
same
has been
side.
raising his hand. Stefan.
Yes, thank you again, dear KTH colleagues. It's nice to have this session with you. I wanted to know, will there be a publication of the recording of the conferences, or I see
I'm gonna pause the recording.